So, in welcoming you here in this very nice location in hospitable Russian land, I would like to thank the IFPMA, the International Association of Pharmaceutical Manufacturers and Associations in Geneva, our senior brothers and sisters, for the idea to hold in Moscow the first regional scientific and practical conference dedicated to biotherapeutical medicines, modern challenges, cultural practices, and approaches for harmonization. And it gives me particular pleasure to thank the Ministry of Healthcare of the Russian Federation and personally our Minister Veronika Igorevna Skvortsova for the understanding, for her support and trust, and for delegating some of the leading experts of the healthcare ministry, as well as their departments, for participation in this conference. I would also like to say thank you to Roszdravnadzor, to the Russian Academy of Medical Sciences, to the Eurasian Commission, to the President's Office, to the Executive Government, the State Duma of the Russian Federation, for their intellectual collaboration, support, and being so proactive in our work. And of course, my thanks also go to the moderators and to the speakers. In many ways, they're wearing two hats, and these actually come from a number of countries of the world, and they're prepared to share their knowledge and their experience in such a sensitive, important, and at the same very challenging area of activity. And my particular thank yous go to all of you, the participants of this conference that come from many walks of professional careers. We have representatives of the medical community, the patients community, we have regulators, we have experts, we have both domestic and international pharmaceutical companies representatives. We also have mass media representatives. All in all, for the participation of this conference, we have registered more than 400 participants. And of course, without you showing the interest to this conference and to the topics discussed here, this would have never happened. And finally, my thanks go to the employees, the limited staff of IPM, for the implementation of this idea, for the design, and for organizing this conference here in association with our partners from IFPMA. With that, ladies and gentlemen, let me tell you that, as I see it, as of now, from the very beginning, the organizers of this conference have every right to say that this conference is a success, in the broadest sense of this word. And this is all that matters. This conference can be called unique and unprecedented in terms of its professionalism, in terms of the depth and the breadth of the topics discussed, and the composition of participants. According to our data, the majority of people in this room have some sort of a background or scientific degree, and that is telling us tons about the authoritativeness and representativeness of this forum. The topic of this forum, besides everything else, is also very important for the global healthcare, but even more so, it is important for the modern medicine and the pharmacy of the Eurasian region, which is currently undergoing a milestone change and improvements in the legislation based on the world's best practices. Let me tell you that the organizers made every step to make sure that every discussion is interactive and deeply involving. And we're absolutely sure that you will be as enthusiastic about it as we are. So, dear ladies and gentlemen, the Ministry of Healthcare of the Russian Federation was supposed to participate personally in the opening of this conference, but due to being so busy, she couldn't make it on her own, and still she was sending us a letter, the official address to the participants of this conference. So, and I'm going to give floor to Head of Department at the Ministry of Healthcare, Sergei Trishrazmov, for reading this address. So, ladies and gentlemen, I'm welcoming all the participants of this forum, and let me read off the welcoming address of Veronika Skvortsova. To the participants of the Regional Eurasian Conference dedicated to biotherapeutic medicines and approaches for harmonization. Dear ladies and gentlemen, dear friends, on behalf of the Ministry of Healthcare of the Russian Federation and on my personal behalf, I'd like to welcome all of you here at this first Eurasian Conference. Holding of this conference is the first attempt of this kind that involves very deeply important issues related to the modern healthcare. Development of biological products has laid a new foundation for treatment of difficult diseases, and a lot of patients across the globe now have hope to cure their cancer, immunology, orphan, and other types of related diseases. We've accumulated vast experience in terms of regulation that calls upon a new, specialized, and dedicated approach and the way we treat this kind of products, including their production, development, registration, and providing quality of pharmacovigilance approach. It's all about bringing together the government, the scientists, the community for one purpose: to treat the patient using state-of-the-art and best-in-class solutions. And I'm absolutely hopeful that getting some of the best practices here in terms of biotherapeutic medicines will definitely help the participants of this conference to not only improve their professional level, but also contribute greatly to the overall healthcare situation in Russia. Minister of Healthcare of the Russian Federation, Veronika Skvortsova. Thank you very much for your attention. Thank you, Sergei. Thank you, Veronika. And now I'd like to pass the mic to our co-moderators of this first session. These are Ivana Knezevich from the World Health Organization. She is heading the Department for Control and Standards, and Madam Maximkina, who is heading one of the departments of the Ministry of Healthcare. Ladies, please take it away. Ivana, you're absolutely welcome. Please.
Good morning, ladies and gentlemen. My name is Ivana Knezevich. I work for World Health Organization in Geneva, and I have a great honor to chair this session together with Dr. Maximkina. So we'll try to co-moderate and arrange the session in the best interest of the audience. First of all, I would like to thank the organizers, the IFPMA and IAPM, for setting the scene for this important event, which, according to the number of participants, has been actually waited for a long time. From WHO perspective, this is very important event as well, because we haven't been able to reach this audience in the past 6 years while we were working actively in the area of biotherapeutics. So this is really a great opportunity for all of us to share experience, knowledge, and I think it's really wonderful that we have such knowledgeable people in the audience, but also around the table. So I think my task today and the task of Dr. Maximkina will be relatively easy. We have excellent scientists around the table, and we'll try to cover the topics as they go along. In addition to the organizers, I think it's very important that we also have high-level officials from the Ministry of Health today with us. And this is also an important opportunity for all of us to call for help and maybe to raise awareness of how the role of regulators is important and how much support regulators need in paving the path to safe and efficacious products in each country. So Purpose of the session is really to open discussion and to call for action. And every one of speakers will highlight some of specific issues and will basically try to come up with some proposals and suggestions. And what we learned from other countries we'll also share with you, and it's obvious that without support from the Ministry of Health, from many other authorities, there is nothing that can be achieved. Regulators alone cannot do much. They can do a lot, but they cannot achieve a lot. And this is a big difference between the two. To help developers of these important products, it's also an important issue that regulators and manufacturers or product developers have a proper dialogue all the way through the life cycle of the product. So it's not just the time point when the licensing comes, but it's from the early stages of the development until post-marketing surveillance and throughout each stage of regulation. So the regulation of these products is the main theme, and we'll try to point important issues, and we'll definitely call for help, and we'll call for your proposals on how to improve the situation, how to work together, and how to make sure that the existing networks in the world can actually help each other, because there are different regional perspectives and different issues that we need to deal with. So this is just a brief introduction of the session. So, as a matter of introduction, I'll just mention that I work as a scientist in the Essential Medicines and Health Products Department in WHO, and you can see the famous abbreviation EMP on the screen. And one particular issue that I've been leading for about 13 years is the area of biological standardization. So I'll give you a brief overview about our activities in the area of biotherapeutics, and we'll then move to some other issues regarding the biosimilars. From the perspective of WHO products and activities, I believe that many of you may be familiar with international standards, and particularly we call them guidelines and recommendations, which I'll show in a minute. So this is considered as a written standard. We also have measurement standards, and these standards are called international standards and reference preparations. And in the area of biotherapeutics, it's very important to distinguish them, because these standards are not reference products. So we'll come back to the point of clarification. I'll then mention our experience with the implementation workshops, and we'll talk about technical assistance that we've provided previously and also what we plan to do next. And then we'll come to the core part of the presentation, and these are the challenges in evaluating similar biotherapeutic products. So having the industry and regulators and other experts, hopefully from high-level authorities in the Ministry of Health to patients' organizations and everybody who is involved in one way or another, will be a good opportunity to highlight challenges in this area of the world for which we don't have much information. So I'll mention also our focus in 2012 and 13, and we'll then move to the regulatory risk assessment of biotherapeutic products. And I have a suggestion for a couple of points for discussion, but it will be open to speakers and audience also to comment on that. So this is just an illustration. What we call written standards is basically the blue book that you can see on the left-hand side. And these are the technical report series. So this is the main publication that we use for publishing guidelines and recommendations for biologicals, for vaccines, for a number of products that are actually included in the context of health products. So basically, number of recommendations and guidelines for broad range of medicinal products are published in technical report series. From the perspective of measurement standards, you're also familiar, especially those who had a chance to work in the laboratory and to do some quality control testing, that the international standards provided by WHO serve as primary standards, and they are used for calibration of secondary standards or working standards that are used by national control laboratories, but also manufacturers. So this is the flow of using the standards, and the value of international standards that WHO provides is in the fact that it provides international unit, which is well-defined unit for potency or for clinical performance. But these measurement standards are actually mainly used for potency and for in vitro tests. So they are not actually, they are not intended to be used for any other purpose, not in particular not as a reference product. So we consider that development of both measurement and written standards requires a lot of science behind these developments. So scientific evidence is the key for the development of both. And many of you who've been involved in consultation process of WHO may remember that in certain cases when we didn't have a chance to find scientific evidence to 100% as a basis for the establishment of standards, we had to go through the consensus. And this is a very important issue to understand, because consensus-based principles are fine, but they may be subject of more frequent changes than those that are based on clear scientific evidence. And usually scientific evidence is coming from the studies that manufacturers are conducting or from collaborative studies that WHO collaborating centers are conducting. So the leading WHO collaborating center for measurement standards is NIBSC in UK. And there are also other collaborating centers that contribute to that process. So we'll be basically talking about science-based regulation, and it's important to understand at this slide that science-based regulatory standards are the best way to go, because they provide really solid assurance of safety and efficacy of these products. So everything that I'm not able to share in 20 minutes presentation you can find on the website, so we'll move on. So this is just to give you the link to the source of information regarding the measurement standards, and especially for biotherapeutics. And this is a brief illustration on the reference preparations that were developed in the period 2008-2012. So you can see that lots of various reference preparations for products like insulin or different EPO products or interleukin or TGF. So all these biotherapeutics are basically covered with reference preparations. What about guidelines? So when we talk about the guidelines on similar biotherapeutic products, here is a quick link where you can find the information easily, and there is also a web page that covers broad range of biotherapeutic products. And one important source of information where you can find number of issues that I'll be talking about when sharing experience from different parts of the world is the link to special issue in biologicals. So following one of the implementation workshops that we held, we actually published a special issue in biologicals, and there are 25 articles with WHO experience in various countries. So you can actually see what's going on in Thailand, in Singapore, in a number of countries, and you may find some sort of common issues in the establishment of national requirements. So something that everybody is struggling with is already available. So what is the intention of WHO written standards? So when we talk about guidelines, how can you use them actually? So they are considered as technical specifications that help define safe and efficacious products. This is the main intention of these products. They are also intended to be scientific and advisory in nature. But advisory part is a little bit problematic because usually the experts involved in the development of these standards would like to make general principles and statements that will not change very frequently. While the users of these standards, regulators and manufacturers in the first place, they would like to see as many details as possible. So the famous example that we show is that when people are reading these documents, they would like to see in the requirement for clinical trial how many subjects are needed. And this is the advice that we cannot give, because whatever advice you give, it will be wrong. Clinical trial design is a very complex issue and requires a lot of scientific considerations. What is the intended use of the product? What are the characteristics of the product? So you cannot actually say that 60 subjects is enough for testing safety in Japan, for instance. But that's the history of setting some of the national requirements. And it's important to understand to what extent advisory role can actually go. So it can go to the point that we have scientific justification. And the rest has to be defined by regulators in the country. So that's why we underline the importance of having expertise in the countries that are dealing with these products, so especially in the countries where development of these products is going fast. So since they serve as a starting point for national requirements as well as a basis for vaccine pre-qualification, this is not the case in the area of biotherapeutics. So pre-qualification for biotherapeutics is not in place for a variety of reasons, but this is something that has been actually discussed in the past years as well. And one of the purposes of WHO guidelines and recommendations as written standards is also to facilitate harmonization of national requirements in the world. So if every country, and we have 194 member states, if every country would follow WHO guidelines and recommendations, there would be a very easy job for both regulators and manufacturers. However, there is not enough details in these guidelines. So that's why the importance of setting national requirements in line with WHO guidelines and recommendations is the first step. But then additional national requirements may be needed as well. So we also consider that these documents are living documents, especially some of them who may require more frequent changes. So the evolving concept of these standards is that they actually started as quality specifications. They started as 2.5 international units for rabies vaccine. There was a very clear technical specification, did not require any implementation workshop. Everybody in the world was actually familiar with that, and there was straightforward implementation. However, today, when we talk about quality assessment, non-clinical and clinical evaluation, it requires a lot more understanding on the overall picture and regulatory requirements. So the concept of the guidelines on similar biotherapeutic products is to provide key principles for evaluating these products as a basis for setting national requirements, but to leave enough space to regulatory authorities to formulate additional or more specific requirements. So while you are working on the guidelines on these products in the Russian Federation, you may actually find some national requirements that you would like to add to the principles that are already provided in WHO guidelines. And for us, it will be extremely important to understand the reason for these requirements, or if there is any discrepancy, if you would like to modify one of the principles that we offered, to understand why do you need that modification and what is the intention. And I'll show you later what happened in a number of countries that we actually work with. So, assisting with the implementation of the guidelines into regulatory and manufacturers' practice is one of our roles, and there are global and regional networks that already exist, but there is also a need for setting national workshops. So we had one specific discussion in countries like China, where a number of products are under development. So certain countries really require additional support. There are also trainings and advisory groups, and the development of these documents always involve consideration of guidelines issued by other bodies. So in the case of similar biotherapeutic products, when we actually started development of WHO guidelines, EMA already had a number of comprehensive documents. So we actually did try to align as much as possible with the European standards, and I think we actually achieved that in majority of principles. So these are just reminders. I'm sure that since guidelines have been in place for years, I'm sure that everybody in the audience knows what the scope and key definitions are. But for the sake of translations and maybe better understanding on what the whole conference is all about, I'll just show that basically the scope is applying to well-established and well-characterized biotherapeutic products such as recombinant DNA-derived therapeutic proteins. And due to the fact that special considerations are given to vaccines and plasma-derived products and their recombinant analogs, they are not considered as part of the scope. So there are different documents for these type of products. When we talk about similar biotherapeutic product, we consider that this is a biotherapeutic product which is similar in terms of quality, safety, and efficacy to an already licensed reference biotherapeutic product. And this is a very important statement. It looks like a very simple thing to do, but in reality it's not. It requires a lot of understanding, a lot of knowledge, a lot of consideration in choosing the right reference product, and then demonstrating similarity in terms of all three aspects. So the reference product is used as the comparator for head-to-head studies with a similar product in order to show similarity in terms of the quality, safety, and efficacy, and only an originator product that was licensed on the basis of a full licensing dossier can serve as a reference product. So again, it does not refer to measurement standards such as international pharmacopoeia or national standards or any reference preparations. So hope that if this part is clear, then the rest will be a subject of details. So the key principles are that similar biotherapeutics are not generic medicines, and many characteristics that are associated with the authorization process and marketed use of generic medicines generally do not apply. The effective regulatory oversight is actually critical for assuring safety and efficacy of these products. And what is recommended in WHO guidelines and in many other guidelines that are currently available is stepwise approach. So the first step is to demonstrate similarity in terms of quality parameters, and this is a prerequisite for moving on with the evaluation of non-clinical and clinical performance of these products. So the most important part in clarifying what is biosimilar versus what is not is the difference is the actual analysis and interpretation of data that were generated through the comparability exercise. And what we repeated in many countries is that if major differences are found in the quality, non-clinical and clinical studies, the product should not be considered as similar, and therefore other options for its further development and licensing should be considered. So what is important to understand at this slide, and I'm usually in the workshops in many countries, we are not moving forward before people confirm that they understand that. And what needs to be understood is that if the product is not similar, it could still be a very good product. So there is no need to claim similarity. If it's different, it's different. It could be good, it could be used, but it cannot be called biosimilar, or it should not be called biosimilar. So linking the name of the product with the actual regulatory pathway is one of the major tasks that regulators have. So if the comparability has been conducted and similarity was demonstrated in terms of quality, non-clinical and clinical performance, in terms of quality, safety and efficacy of these products, and it's confirmed as similar, then the product should be named biosimilar. Otherwise, it can be done in a different way, it can be regulated differently, and can be used if it's demonstrated as a suitable product for the intended use. But this is the major issue in a number of countries, that the initial intention to demonstrate similarity goes to the point that if it's not similar, it's still called similar. And this is creating a lot of difficulties for both regulators and manufacturers. So this is a very simplified approach that many countries follow, but in reality, the picture is a bit more complex. So what is considered as a pathway for biosimilars is in the middle, in green color. And it shows that it's based on existing knowledge, full comparative characterization plus comparative but reduced non-clinical and clinical data, and it's applicable to well-characterized biologicals only. So they should not be regulated as generics, and they are, the regulatory pathway is different from what is usually used for standalone approach, which is also applicable to all biologicals. So many innovative products are going through standalone approach, but there is a specific problem with that, which I'll point out in a minute. So the reference product, which I already mentioned, is just to underline that the experience with the use and selection of reference product is different in different countries. And this is a big issue that I'm sure many speakers will point out. From WHO perspective, it is acceptable to use reference products licensed in other jurisdictions, but apparently WHO is just providing and offering guiding principles, and national regulatory authorities have to decide what is the most suitable for their regulation. So in some countries, reference product must be licensed in their countries, and this is the case in EU. While some other countries, like Canada, which is a huge country but relatively small market for biotherapeutics, they accept products licensed in other countries. Of course, there are certain criteria, but there is a kind of flexibility. So when choosing the right way for your country, it's good to know how the others are doing. We are going to have detailed discussions on quality, non-clinical and clinical. So this is basically just a quick reminder what is in the guidelines and what you can consider as a principle. And basically head-to-head comparison is the key. So I'll go quickly through some of these principles for which I believe that are well understood, and then we'll focus on the experience in the countries. For non-clinical evaluation, everything that you can find in the guidelines is just briefly summarized here. You have in vitro and in vivo studies. You have discrepancies in the experience with these studies. In some countries, there is a kind of insistence of head-to-head comparison in each aspect of non-clinical evaluation. In some countries, there is a bit more flexibility in conducting these studies as comparable study, so you'll actually find a variety of different approaches, but it's important to pay attention to the interpretation of this data. So for clinical evaluation, which is actually a highly complex issue, and comparable safety and efficacy of the similar product to the reference product is the key. You'll find the details about the efficacy studies, about safety evaluation, about extrapolation in the guidelines. And I just like to mention that extrapolation is actually extremely complex issue, and it depends on the number of factors. So it's really important to deal with the indications and to understand what is the intended use of the product. Another short section in the guidelines where you can actually find really key elements regarding the pharmacovigilance is basically saying that close monitoring of the clinical safety should be put in place, and the focus on serious adverse events in all approved indications is important. To identify SBP and to have traceability, it is actually suggested to use brand name, INN, where it exists, lot number, and country of origin. And one particular problem that we found, for instance, in Thailand with the EPO product was that very limited information on the product was provided, and it was impossible to trace back to the product which was actually used. So that's why the whole issue of safety monitoring becomes very complex if traceability of products is not possible in the country. So pharmacovigilance system should be in place at the time of marketing authorization, which is easy to say but difficult to make sure that that is really in place. And also it's recommended that manufacturers should submit a safety specification and pharmacovigilance plan at the time of submission of the marketing authorization. And I just want you to remember that this is really a unique opportunity during the licensure to agree on the pharmacovigilance plan and to make sure that additional data that are not available during the licensure will be provided in the years to come. Because in particular, when we talk about safety, data package is limited. So you cannot do much for licensure. Even if you try hard, it's always limited information. So the best knowledge about safety of the product is coming post-licensure. But if you don't put the plan in place, you will miss the opportunity to learn about these products. So that's why we keep saying that number of years for which the product is in the country doesn't mean a lot. But the monitoring means a lot. So you can have product for 2 years and no more than if you had product for 10 years, but you didn't actually have a proper surveillance. There are also risk minimization measures that may enhance safe use of biosimilars, and NRA should actually monitor compliance. So these are kind of general principles that are there. The International Conference of Drug Regulatory Authorities in 2010 made recommendations to countries on how to deal with these products. And one of the recommendations is to implement WHO guidelines as a whole. Not just part of the guidelines that say, well, we follow the principles for quality assessment, but we have something different for clinical. In that case, we cannot say that WHO guidelines are implemented. Strengthening of expertise is also recommended, but there are also recommendations that IGDRA made to WHO to consider development of guidelines on risk management strategies for copy products licensed as biogenerics, to develop template to help member states to share information regarding the scientific basis for licensure, to supplement guidelines with the guidelines for biotherapeutics in general, and to conduct review of existing reference preparations. So most of these have been actually done. So what I'd like to mention here is that implementing WHO guidelines without modifying principles is good. With modifications is also acceptable because responsibility is at the country level. Responsibility is with regulators. So they have full right to modify the principles and to do something different. The only plea that we have is to let us know what the modifications are due to and why these modifications are needed. So it will be good to hear that from your development of guidelines and products as well. A few points from the implementation workshop in Korea in August 2010, when we actually reviewed the situation in 12 countries. And basically, one of the important issue, since these details are all published, and you can find the information about that, it was actually clear that experience with comparability study is very limited in developing countries. And it's difficult to understand how comparability study, for the purpose of demonstrating similarity, should be conducted, and also how to interpret the data. So this is one of the challenges that I actually included here. Couple of examples that we found that, for instance, partial comparative data inequality were done. And then comparison with literature in the context of clinical comparison. This is, according to WHO principles, is not acceptable. So if comparability studies are done, they need to be done head-to-head with the reference product. Comparability with historical data is a valuable additional information, but cannot replace head-to-head comparison. So all kind of modifications that exist in countries for a reason or another are already in place. And this is something that we would like to improve and probably make more consistent between countries, but it's not an easy task. So the terminology used in different countries also showed that basically biosimilar pathway is not yet in developing countries. This is a development of biotherapeutic products which have gone through different assessment, but we cannot say that this is a real biosimilar pathway. We also had a workshop in China last year, and we were focusing on quality assessment. We found a number of issues for follow-up, and we also found that certain principles need to be discussed further. And one kind of strong recommendation was not to use the term biogeneric because it's confusing and it doesn't really give a good explanation what these products are. So there was a call for technical assistance in the workshop in China, and we are still dealing with a number of requests for preparing questions and answers, developing e-learning tool, and providing broad assistance to regulators in the world that are actually trying to improve their expertise and to build their capacity. Now, the key point for our discussion are the challenges. So we found that regulatory framework for biotherapeutics has a diversity of approaches in the world, and that most of biotherapeutic products in developing countries belong to the category of known biological entity that undergoes through the standalone pathway with reduced data package, rather than through a biosimilar pathway. So many countries reported, I think EMA also has that possibility that something that is known biological can go through standalone with reduced data package. But then the question on to what extent that reduction can be done is always questionable. So if you ask EMA, do you have guidelines on that? They would say no, because our experts know how to assess, and they would give a kind of scientific opinion on that matter. But when you go to developing countries, they would like to see something in WHO guidelines that can actually help them defining to what extent reduction of data can be done. So some countries also reported that they have non-innovative biotherapeutic products, which is based on evaluation of these products with reduction of clinical data usually, but these requirements are not always clear. And there are a number of issues that are going beyond regulatory responsibilities. So that's why I think that initiating discussion at the national level with all relevant authorities, including regulators, but also other relevant authorities, those who are dealing with patents, those who are dealing with interchangeability and substitutability, labeling and prescribing information. This is something that we were not able to include in the guidelines, but our recommendation is that regulators need to be involved in these discussions, and even if it's not their direct responsibility, they should be informed on what are the decisions and what is the basis for decision in the country. So our focus in 2012 and 13 was on these implementation workshops and also on the revision of the guidelines for products derived by recombinant DNA technology. And this is the document that we plan to submit to the Expert Committee on Biological Standardization in October this year. So we had first round of public consultation in March, and I believe that many of you had a chance to review the document because we specifically asked IFPMA to share with its members. So we believe that at least industry received that. With respect to regulators, one of the purposes why I'm here this week is to find focal point and regulators who can actually work closely with us, because this is the audience that we were not able to reach in previous years. So this is something that you should be able to review in the second round of public consultation if you didn't have a chance to see this beforehand. There will be further discussion with Latin American countries in September, and this is a very strong network of regulators that is highly interested for biotherapeutic products, and this is not the interest of yesterday. They've been actually asking for assistance for years, so this is coming next. There is also a very short and brief guidance document that is under development and is talking about regulatory risk assessment for biotherapeutic products licensed with insufficient or inappropriate data. The issue is that in the years when clear guidance have not been made available or have been subject of different sort of level of acceptability by different countries, products licensed at that time, probably without clinical data, were considered as product license with insufficient data. And this is an issue that we would like regulators to deal with. So instead of saying, no, we don't have these products, it's better to say, yes, we do have these products, and we are going to take action. So the pilot study that is going on in Thailand will hopefully stimulate other countries to take action as well, because they realize that certain products were licensed without sufficient data. We all recognize that it's not their fault because the information was not available, it was not well understood and accepted what needs to be done. And now the question is, what is the option with these products? So the options that you see on the screen are subject of discussion, so don't take them as a current approach. This is basically subject of revision and will definitely change that. But basically there is a possibility for leaving product on the market and then strengthening post-marketing surveillance to identify possible adverse events associated with their use. There is also a possibility for withdrawing a product from the market if there are certain safety or efficacy problems that have been identified. And there is also an option to leave it on the market for a specified period, such as 4 years, during which time manufacturers would be required to submit appropriate data that were not submitted originally. Option number 2, withdraw from the market immediately, is not recommended because other 3 options give enough consideration regarding the potential use of the product. So these are basically the issues that are subject of discussion. What I'd like to suggest that we focus on in the discussion time after all presentations is to deal with the national regulatory requirements, basically to consider the situation in Russian-speaking countries, because this is something that is now subject of further discussion and development, to consider what are the regulatory requirements in place. For instance, Is there non-innovative pathway, or what are the options for regulation of biotherapeutics? And I believe that speakers in this session will address that point. To focus also on the guidelines on biosimilars in this part of the world, and also to see what are the products licensed in Russian-speaking countries that are considered as biosimilars or maybe biotherapeutics with some sort of intention to be similar. And then regarding the experience and information sharing, we are promoting the concept of science-based regulation, so that requires expertise and capacity building. So it's important to discuss how that can be improved. And there are a couple of examples regarding the information sharing. So our general recommendation is that regulators should be as transparent as possible, which means that regulatory requirements should be published on their websites. Manufacturers should be very clear on what they need to submit and what they need to do for certain products at certain stages of regulation. And also, following licensure, it's also important to increase transparency by providing the information on what was the basis for licensing these products. So the example that European public assessment reports provide, or Health Canada also has public assessment reports, TGA. So is there a possibility for having such reports issued by regulators in Russian-speaking countries? Even if it's in Russian, it would probably stimulate some of us to learn Russian while the translations are going on. So, final point for discussion is what is most needed in terms of technical assistance. So I'll stop here and we'll basically go through the rest of the session with the other speakers. Thank you very much for your attention.
Thank you very much for your kind presentation, and hello to everybody. And first of all, I would like to thank the organizing committee for the kind invitation to be here and to give this presentation, trying to explain the key issues on biosimilar guidelines at the European level. This is my address, my name, and I'm the Italian delegate at the Biologics Working Party at the European Medicines Agency, and I've been appointed by the Italian Medicines Agency, AIFA. This is the disclaimer. According to the European Union and EMEA policy, I have to declare my conflict of interest according to the EMEA document which is mentioned on the presentation. I know that we are all familiar with this basic concept, but I need to give a very brief introduction on what we are talking about, at least from my point of view. Well, we all know that biological and biotechnological products are quite complex molecules, so they are not easily characterizable from the biochemical and immunochemical point of view. We also know that the characteristics are essentially linked to the manufacturing process which is developed by individual companies. So this is something which, in my opinion, has to be taken in great account. And this is also the reason why the generic concept, which is quite applicable for chemical entities, which are small molecules were characterizable from the biochemical point of view, is not applicable to the biological product, and this is the reason why it was introduced, the term biosimilarity and biosimilar product at the level of the European Union. This was done, I'm not going to go through the text, but this was done essentially when the Directive 2001/83 as amended was approved in Europe, and it was also clearly said that when products have biological products have different manufacturing process, may use in their process different raw materials and are obtained from different starting materials, well, they cannot be considered chemically identical, and the results, as it is written in the directive, of appropriate preclinical and clinical studies are required to prove this biosimilarity. So the concept of biosimilarity is well embedded in the European Union legislation. Therefore, what can we do now to prove biosimilarity? What is actually the process leading to the biosimilarity definition and assessment? First of all, the biosimilar product should be fully and extensively compared with the originator. And this is something that has to be done at various levels, mainly at the quality level, but of course we also need a comparability exercise carried out at the non-clinical and clinical level. We will learn a lot about this approach also from the speakers who will follow me during this morning session and also in other sessions. As I have said, the comparability approach, which is essentially called the comparability exercise in Europe, should be start as soon as a company has decided and has set the idea of developing a biosimilar product. In fact, it's quite clear that if you start with a comparability exercise quite late, when you have already partially developed your product, you may end up, if at the end the results are not supporting the biosimilarity, you may end up with a number of non-clinical and clinical useless studies, which means also use of animals and use of patient and performance of poor quality clinical to useless clinical trials. So the main focus is the comparability exercise, and what is quite curious, at least in my opinion, is that this comparability exercise approach was not developed to prove biosimilarity. Actually, the origin of the comparability exercise is linked to the development, to the evaluation of changes introduced in a given manufacturing process by a given manufacturer. So the comparability exercise was first developed to control changes within one manufacturer. So we're not talking about comparing products between two manufacturers, the originator and the biosimilar, but within one single manufacturing process and one single manufacturer. And this comparability exercise is based on a repeated and extensive testing of the drug substance and drug product obtained in that specific case before and after changes introduced in the manufacturing process, but in the biosimilar case approach between the originator and the supposed biosimilar product. Of course, the European Medicines Agency is deeply involved in this process, and it has been already said by Ivana, that by my colleague, that the importance of sharing approaches throughout the world is quite an important issue which should be somehow discussed. At the European Medicines Agency level, we have a clear approach, at least in my opinion, it's quite a clear approach. Biosimilars are evaluated according to the centralized procedure. I don't know whether you are familiar with that procedure, but all biotechnology-derived products are assessed by the European Medicines Agency. There are a number of documents which have been issued by the agency to help applicants or regulators to deal with this topic of biosimilarity. And especially we have documents and concept paper dealing with general issues like defining the overall policy and, of the biosimilarity approach, and this is an overarching guideline. We will talk a little bit about it later. And we have guidelines which are module specific. I'm referring to the concept, to the definition of module according to the CTD ICH format. So we will have guidelines on quality, non-clinical and clinical sections. And we also have a lot of specific guidelines which are dealing with a specific product. Then we have a process in which the dossier is examined by the European Medicines Agency, and the quality part is assessed indirectly at the level of the Biologics Working Party. There are, of course, classical steps in the centralized procedure I'm not mentioning here, but the final quality assessment for this type of product, also for recombinant products, is then carried out and harmonized at the Biologics Working Party level. There is also an ad hoc working party on biosimilar medicines, which again has been created at the European Medicines Agency level to specifically look at this problem of biosimilarity at the clinical level. But the final decision anyway is taken in Europe by the CHMP and finally endorsed by the Commission. The one point which was mentioned by Ivan is quite interesting, and I would like to stress it. At the end of the process, in Europe, we have a European Public Evaluation Report, assessment report, in which a lot of information included, although a lot of information, confidential information from companies are also excluded. But it's a matter of how this approach is carried out, and I think it's also a matter of transparency, as it was mentioned earlier. We have several main guidelines, and I would like just very briefly to go through them. Again, we are in a very, I would say, in interim process because we have some guidelines which are not old, but they need to be revised, and I will also talk a little bit about that process. The main guideline which was issued in almost not 10 years ago, but in 2005, it was a general guideline on similar biological medicinal products, the so-called overarching guidelines, because it was a guideline, and it is a guideline covering general issues on this field. And this guideline explains that there are, well, first of all, a few rules to be followed. One is that the reference medicinal product has to be chosen, and this was already stressed by Ivan in the previous presentation, within the European Union, within the community actually. And this means that we must be sure that there was a full assessment in terms of quality, safety, and efficacy of the originator of the reference product, as we call it in this guideline. We may also have supporting data, of course, but the use of this supporting data is not, I mean, it has to be used as supportive data, nothing more. So we need to run a full comparability program with the originator. And again, the active ingredient, there are also a few specific indications concerning the nature of the comparison that I have to make in terms of selection of the originator. And for example, the active ingredient in the biosimilar product must be in as similar as possible. This may look like a sort of a tricky sentence, but it's clear that if I have to make a biosimilar product to an originator which is synthesized which is produced by using Escherichia coli, which is a prokaryotic expression system, I would avoid to go for a biosimilar product manufactured using CHO cell lines, which are eukaryotic cell lines. So this is just the type of concept that are included in this general overarching section of the guideline. The analysis of as I'm reading through the slides, I mean the concepts are quite clear, and also the analysis of finished product, if available, should be also extended in relation to the quality and to the presence of excipient and other components in the finished product, which may have some impact on the definition and on the analysis of the product itself. This has to be done always taking into consideration that, as I already mentioned, the quality of and therefore the efficacy and the safety of a biotech product are defined not only by the quality issues and the quality results that I get after the comparison, but also taking into account the fact that I have a product which is defined also by the manufacturing process, and as a manufacturer of a biosimilar, I am not aware of the manufacturing process used in the originator. As you know, this guideline is under revision, and it's a common policy of the European Medicines Agency, as it is a common policy of other organizations to revise the documents which are now on the table for a while. And the overarching guidelines is one document of under revision, as I said, and the revision was the last draft, which was issued for public consultation, was very recently licensed by the CHMP at the beginning of May, and the consultation period will last till the end of October. The main changes which have been introduced in this new guideline, not new guideline, but in this revision of the existing guidelines, is a clarification of biosimilar approach. An example was what I already mentioned in terms of choice of the originator of the reference preparation. And then there is also a sort of clarification of what we intend to have in terms of supporting data or data coming from a product which was not authorized within the European Economic Area. I guess we will hear a little bit more during these 2 days, but essentially, if we want to use a product which are not marketed within the European Economic Area, we can use this data, but we need to perform a comparability exercise, taking into account not only the originator and the supposed biosimilar product, but also using in the comparability exercise the product which was authorized outside the European Economic Union. And therefore, if the outcome of the comparison is favorable, I can use this supportive data in a very, in a more appropriate way. So it's a way to accept also partially accept data from product obtained, manufactured and approved in other countries. We also have quality guidelines which are defining in better detail the criteria for the process and the criteria for assessing biosimilarity at the quality level. What I would like to say in particular is that, as I already said, in the biosimilarity definition is included the concept of a comparability exercise, and therefore the quality guideline, besides mentioning a few general concepts, as you can see from the slide, and besides mentioning the immunogenicity issue, which is an important point which will be discussed in other presentations either today or tomorrow, there is again stressed the concept and the reference to the comparability guideline, which is an ICH guideline, which talks about the approach to be followed to make this quality exercise and quality comparability with all biotech products, as I have already mentioned. And of course, this is what is included in the comparability guideline. And I would like just to underline a few points. Well, first of all, the demonstration of comparability does not necessarily mean that the quality attributes before and after the change, in this case, or between the originator and biosimilar are identical. It means that they must be similar. So it's a very tricky approach, but we are never searching for identity between two products, either before or after a change, or between an originator and a biosimilar. Another approach, which in my opinion is quite important, is that it is stressed the concept that we have to use a battery of tests, analytical tests, which have to be performed to prove biosimilarity or comparability at the quality level in more general terms. And finally, the other point, I guess, in this guideline is mentioned is the fact that the analytical systems that we use must be state-of-the-art and must have this orthogonal approach, which means that they have to look at the same quality attribute by using different analytical methods. This guideline is also under revision, and it has been already put out for consultation, and we will come back with a new version as soon as these activities are finalized at the EMEA level. In this new text, there is a better clarification of the formulation issue. So I guess that you have already seen this document, and because you are supposed to have already commented on it. There is a comparability section which has been extended as compared to the one which was in the previous version, and so on and so forth. The important point, again, in my opinion, is that although the concept of similarity is still present, the concept of not identicality is again stressed between, in this case, the two products between before and after the change, or the originator and the biosimilar. We also have clinical guideline and non-clinical guidelines, sorry, and the main message I would like to give for the moment at this general level is that according to the outcome of the comparability at the quality level, the main approach will be case-by-case basis for non-clinical guideline, and essentially, for the sake of time, and essentially the same concept applies to clinical guidelines, in which, again, according to the comparability level we will achieve, the quality level we will have to plan for adequate studies. And At the clinical level, there are also a number of guidelines which are specific for each individual product, which has been already authorized or which will be in the future put on the market, just to clarify aspects which are linked to the clinical assessment of the comparability and of the biosimilarity, in this case, exercise. These two documents, non-clinical and clinical, are under revision again, and there is a concept paper. I'm not going to enter into detail. The normal procedure at the European level is that first the CHMP issue a concept paper, and then if the concept paper is approved after consultation, the revision of the guidelines starts. The emerging issues at the level of biosimilarity and biosimilar product, well, it's one of my last points in my presentation. Well, we have for sure the development of more complex biosimilar, which somehow needs some re-evaluation, reassessment of the concept of biosimilar product. From the non-clinical point of view, we have the issue of identify relevant species which may be used in the studies, and this is not easy. We know all the difficulties we have in Europe to use non-human primates for several studies, and this may become an issue in the future. We also have to take into account WHO guidelines, and I guess there will be a good opportunity to share further details after this meeting. And as I already said, the 3R principle, which is replacement, reduction, and refinement of animal use, not only at the non-clinical level, but also in the testing of biological product, will be an important task to be achieved in the future. Finally, critical issues for biosimilars. In my opinion, despite the existence of a number of guidelines, despite the activity of the European Medicines Agency, there are a few issues still pending with biosimilars. And just to make it very short, one of the main difficulties which I guess will be discussed again here today or tomorrow is the fact that at the beginning it was thought that having assumed, having proved a very good comparability at the quality level, then the size of the clinical and non-clinical studies would have been just nothing or quite reduced or from this point of view quite attractive for the manufacturers. I'm not a clinician, I am a molecular biologist, but I was told by our clinicians that actually the size of the clinical studies and of the clinical trials required when you have to prove comparability at the clinical level between an originator and a biosimilar may even be larger than the study which was followed by the originator per se. So I guess that this is an issue which may require further evaluation. But nevertheless, we had at least, I made some calculations, very recent calculations. I was not able to get an official table, but I counted almost 14 already authorized products. And there are more products which are on the pipeline, including monoclonal antibodies, which will represent a good example of a very complex molecule. So these are just the conclusion. I would like to point out only the last dot in my slide. At least in Italy, but I would say also in other countries in Europe, there is the idea that biosimilar products are not as good as the originators. There is the idea that they go through a sort of shortcut approach, regulatory approach, and assessment approach, and so there is the idea that they are not properly evaluated. This is not right, of course. There is no distinction at the European Medicines Agency level between the process of assessing the originator and the biosimilar. They go through the same procedure, and there are no differences at all in the quality of the assessment. And I have finished, and thank you very much. Thank you.
Dear ladies and gentlemen, I'll try to be very brief today. The thing is that my presentation reflects the special regime that the Russian legislation exists in terms of developing rules and regulations with regards to medicines, including the products that we call biosimilars, as well as their analogies. And the thing is that this presentation is a mixed bag. It's partially translated into English. There's also a slide on French, and the rest is all Russian. The majority of the audience here today are actually quite familiar with the way we proceed in terms of regulation. You know the course of activities, you know what we've been doing over the recent years. I'll try to stick to the wording of my presentation because, unlike Mr. Pini, I will not be able to express my personal viewpoint today, because every word I'm going to say will be viewed as the official position of the Russian Healthcare Ministry. So this is what I've learned over the recent months. So to avoid that mistake, I'll try to stick to my presentation and to never deviate from it. So, to inform our foreign partners and colleagues, let me say the following. The thing is that the changes that have been happening in Russia over the past, let's say, 20 years, all these changes, of course, and I'm speaking about changes of political environment and economic environment in the country, and other changes, especially during the early stages, especially in the early stage, had this slump. There was a gap. So we had to develop new institutional norms, rules and regulations. And of course, in particular, these changes were quite rapid in healthcare. It all started with the official national healthcare program that was announced by Mr. Putin, the President of the Russian Federation, in 2006. And here in the slide, you actually have some of the key details, key regulatory mechanisms that were developed starting from 2006 and up to 2012. And the recent key changes that were adopted and has taken considerable impact on the overall legislative activity will be two federal laws. And one of them has to do with the safety of Russian individuals, and the second one has to do with the circulation of medical products. Now, as I've said, the framework of the Russian Federation over the past 20 years with regards to legislation has been characterized by the fact that we are always playing against time. There's always time pressure. And that's probably the reason why we sometimes fall short of some of the international norms. And I'm speaking about the norms set up by the global community. Sometimes we do it only partially. It's never complete from the viewpoint of quality standpoint. So, and that's why, despite the fact that there's been major improvement with regards to the Medical Product Acts of 1998 as well as 2010, and as a result of these two federal laws, the overall position of the Russian government with regards to seeing any medical product as a complete life cycle from design to consumption, there's been problems. So 3 years of practical implementations show that there is a need for a major improvement and amendments to the existing laws. these are very much called on. And especially I'm talking about the last year. Regulators have been very proactive here. So what are the key areas that I would like to reflect here today? What are the novelties that you can find in this new act? Let's look at this new legislation, and let's look at the novelties that are outlined in it. First of all, it deals with the terminology, the definitions, and of course I do understand the position of the developers of this act. I mean Federal Law 61. I have seen some of the early drafts and proposals. I have seen the definitions that were tried to introduce into this act. I mean, such as the definitions of biosimilars and biological products. But unfortunately, being pressed for time, the fact that regulators always need this yesterday, this always scotches our development. But in many ways, that's actually a good point, due to the fact that because we're pressed for time, we can borrow directly from global practices. And let me show you, by the way, the definition that we decided to come up with for this law. I believe that it's much more comprehensive. Maybe it's going to be interesting to our foreign partners. Why so? Well, the thing is that we're actually seeing that there is no unilateral approach towards the subject of the regulation. And that is a huge challenge, because what are we regulating? What is considered as the biological product? Or, I mean, should we be very strict about it, or should we be a little bit more loose about it? Specifically with regards to these products and other types of and classes of medical products. For instance, in Ivana's presentation, I saw today that the WHO considers vaccines as biological medical products, and there were references to that. Another very important point is that, well, it's not, of course, a scope of our today's presentation, but I believe it's also noteworthy and should be pointed out, and should be quite interesting and remarkable for the regulators, is that we're trying to regulate orphan diseases-related products and optimize the registration procedure. And that means that we need, let's say, further measures that will be imposed by the regulatory bodies, and namely the Ministry of Healthcare of the Russian Federation. We will also proceed to yet another development, and that is, I think, is probably the most challenging thing. And I'm talking about lack of standards and recommendations that Ivana mentioned with regards to things like certain classes and subclasses of medical products. I'm not speaking about key classes, but let's say subgroups of biological products. Of course, as I have mentioned, we have this advantage. We can borrow from best practices immediately because we don't have a backlog of our own practices, so we're quite open here. But it's still never easy. With regards to defining the regulation subject, what are we regulating? The challenge that we face there is that there has to be positioning for these biological products. The thing is that in the official law of the Russian Federation on circulation of medical products, the only group there that is currently part of the overall definition is immunobiological products, and nothing else. What we've come up is a definition that brings together two viewpoints, two different sides. First of all, we would like to set out as a separate class of products in terms of quality assurance procedure. And secondly, we decided to also look at these products from the viewpoint of production technology. So what we call biological products, biological medical products, are several classes and subclasses of products, such as immunobiological products, as well as these are also products that are, let's say, blood and plasma-derived, both human and animal. The next group includes products that is a result of biotech, gene modulation, and therapeuticals. Besides that, if you look at the key issues that we have to deal with when developing regulatory norms, rules and regulations, and specifically the conference that was organized here today is particularly interesting and useful in this regard. The thing is that the discussion is not always fair, I would say. There are a lot of references to global practices, despite the fact that, as we're seeing it today and as we've heard from other speakers, and both Ivana and Pini both mentioned the fact that global international practices are not necessarily fully aligned with reality, specifically with regards to these biological products and their similars. So we have these two things to deal with. The definition of comparative set. So how do we select a product for comparison study as number one? And number two is interchangeability of these products. Most people here in the room know that at least twice we've introduced this activity that has to do with helping establishing a relationship between the biosimilar products. Then we sort of said, okay, let's consider that. And then again, based on global studies and best practices out there, also having studied the official requirements of both patients and the community, and based on the risks that we've seen, we see that there's no regulatory system in the world that is prepared to actually take responsibility for that and acknowledge a biosimilar product to be a product that is actually available for interchangeability with the originator product. Besides that, there's been a number of interesting statements made by Ivana. She mentioned this issue of naming of a biological product, about the originality of name, and about the pharmacovigilance of a specific brand name. That is also a very correct approach, I believe. We have this norm, and this norm actually involves the so-called rational approach towards the naming of the product. And I believe that this kind of discussion is very valuable. We'll be engaged in this kind of discussion, and we need that to make sure that there's a stronger link between a particular biological product and its commercial trade name. And that would ensure a high level of pharmacovigilance over this very challenging class of products, despite the fact that they have indeed a high potential for application. Also, from discussions with Ivana, it turns out that the result of this seminar, of our workshop, would be another issue. I'd like to address this issue to all of you. What about the responsibility of the manufacturer for both the quality and the positioning of the product? Because indeed, the patient is always subject to these exogenous factors. There are several driving forces when selecting this or that product. It's not just the doctor that prescribes you the product, not only the healthcare official, but also the manufacturer that currently has the whole array of medical representatives and other tools that inform both communities about the real effects, about the real specifications of the product in terms of safety, efficacy, in terms of the range, the scope of activity, scope of application. And that is a huge responsibility that the manufacturer has towards the people and towards the government for placing the product on the market. As an organizer, I had a sneak peek into the presentations before the beginning of the conference, and let me tell you that the presentations that we're going to hear from Mr. Alexander Mironov answers a lot of questions that I'm posing and that my presentation is supposed to answer, specifically about the direction in which we're moving. So I'm not going to, take the word out of his mouth. So only to, highlight some, items with regards to quality assurance. We have developed this quality control body during the adoption of Federal Law 61. Now, in terms of interchangeability, we are planning this, like I said, as part of this conference. And of course, we'd like to, you to participate. I think that we all should be enthusiastic about it. And hopefully, this will raise the true cause. That is, what is primary in this discussion? Should it be about the economic drive or should it be about the therapeutical drive? So are we acting on behalf of the patient or on behalf of the company? And besides that, I would like to also give the audience the data that are currently available. And you'll be surprised. It's going to be quite surprising for international regulators. Because what you will see in this data is the list of products that have been actually registered as biological medical products. The data will, of course, range and differ a little bit from Alexander's data, because this table also includes not only the names, but also the manufacturing locations where this or that particular product was registered and is currently being produced. And they all part of the government official register. So my colleagues will be speaking about specific items, but what we carry over is the whole register of these products. So if you look at the total number, total quantity of these products in Russia via the globe, we see that many of these biological and medical products are currently being produced here in Russia. And as Ivana said, WHO calls upon, as a regulator, of course, on a global scale, to actually assess the true, let's say, correctness of a registration procedure, whether the products registered were registered correctly. And a couple of more slides here today, since Alexander did not mention in his presentation this fact, so I believe that I should do it, and I believe that's something that you should know. We're going to establish a central expertise center, and this expertise center will assess the medical products as well as pharmaceutical substances. There are several locations around Moscow. All of this activity will be based around Moscow, and we have a legislative act that has empowered this particular body, and our colleagues will be doing all the details and work. We've defined the organizational structure of these bodies and the whole scope of expert activity that will be conducted by this body. And there was also a slide in this presentation, and Alexander will also mention about that, and I think it's going to be an interesting discussion on that. So specifically, I'm speaking about the pharmacopy standards. There definitely has to be standards, and what we say is that there should be a common pharmacopy approach towards immune products. Finally, let me give you the statement that was actually announced by an American physician, former colleague, of course. In this world, it's not about where you are, but the direction in which you go. So I think that this conference will push us in the right direction, and we will no longer be standing still on the crossroads. Thank you very much for that.
Здравствуйте, уважаемые коллеги.
Good afternoon, dear colleagues. And to begin with, I would like to express my gratitude to all of you for the opportunity to be here and to share our experience about the registration of biological products, including biosimilar products. To begin with, let me say that this presentation today is quite brief. Our intention was to actually give you a sneak peek into our very limited experience, because this is what we're doing now. We're registering these products. On this slide, what you see is the pathway. This is the way we register any product, be it biological product, be it a non-biological product, doesn't matter. So there is a registration dossier, and this dossier is submitted to the National Center for Medical Expertise as well as medical equipment expertise. So this is a governmental body, it is an expert community represented by the Ministry of Healthcare. So our national center is located in Almaty, which is the southern capital of Kazakhstan. The main purpose of our organization is all about running expert analysis of the registration dossier, including efficacy, potency, quality, and pharmacovigilance. Now, once we've done that, once we've fully assessed the registration dossier, we then write a comfort letter. So, during the final conclusion, the regulatory body, which is Committee for Control over Pharmacological Activity, under the auspices of the Ministry of Healthcare, that is also located in Astana, which is the capital of our country, then issues an order to either register or to actually ban this or that product. Now, what I'm going to focus on today are some of the milestone things that we do. These are key departments of this body that are involved in the assessment of quality, efficacy of the product. We have a dedicated department that deals specifically with medical products or medical tools and medical equipment. So each department has both the so-called primary expert opinion, which has to do with the assessment of registration dossiers. So we look in terms of all the necessary documents and the like, and down to, of course, analytical department. Analytical department looks at the quality issues and the quality of the medical product or a medical tool, vs its application sphere, or vs certain norms that we're guided by in terms of this or that product. And of course, we possess laboratories. We have laboratory of microbiology, physics, and chemistry, all the necessary equipment. We got the Republican Immunobiological Laboratory that was established back in 2008, specifically for that, for the assessment of biological and immunobiological products. We also have the Pharma Laboratory, where we test for pharmacological products, including bioavailability and biosimilarity. Since November of last year, we've also established yet another department dedicated to both preclinical and clinical studies. Why did we do that? The thing is that with each year, we're seeing more and more research around products that are both domestic and international ones. And the domestic manufacturers of products are willing to go innovative. And for these types of innovations, we've established this department. This is a support group that helps producers develop new innovative products, as well as, methodologies, guidebooks, regulations on that. Besides that, we also had yet another department that deals with the assessment of quality and safety of this or that product. That's pretty much a certification department. They certify those products that have been registered and imported into the country. We also have 11 territorial branches that are located throughout the country, and these ones also deal with the registration and certification of imported products. Now, let's have a look at the website. This is the official website of our national agency, where any producer can submit his application and then track it, look at the status of this application, look at the certificates that were issued with regards to their products. And besides that, on this website, you can also track down information related to pharmacovigilance, such as any cases of side effects found either in our country or somewhere abroad. So there's a lot of benchmarking involved. Now, the key requirements towards the registration of these products are set out in the code. We have the so-called Healthcare Code of Kazakhstan, and that was established back in 2009. And besides that, we have the official decree number 731 and 5736 about the registration of products and also about the expert assessment of the registration dossier issued by the Healthcare Ministry. Now, during the process of this registration, we go through 4 stages. The first stage, like I said, there is the initial expert opinion. There's also an analytical stage that is more advanced, where we look at specific samples that are given to the laboratory, where we put them through rigorous testing. We have the full body of analytical requirements based on best practices and recommendations of producers, as well as requirements of local pharmacopy of our country. Besides that, we also look at the assessment of this dossier from a pure pharma business standpoint. We have a dedicated department that deals with pharmaceutical expertise, that collects all the documents and double checks all the documents that are part of the dossier, and they look at it specifically from the viewpoint of procedures, production processes. They look at the quality of the mixture, side effects, its stability, look at additives. And another stage is the pharmacological expertise that involves both preclinical, clinical trials, and also the pharma control, as well as risk assessment and control plan. According to Decree 735, the list of documents that are supposed to be submitted as part of the dossier is set out in Module 1. So there's a list of official documents that are subject for this process. We're predominantly dealing with risk management. Now, in terms of pharmacovigilance, in terms of risk control, there will be the next presentation that will dwell in more details on these issues. What about biosimilar products? How do we regulate that? Before 2009, since we didn't have any healthcare code before 2009, there were 0 requirements in terms of registration of these biosimilar products. And what that means that in collaboration with the pharma business, with the Association of Pharma Companies, with the Association of Patients and the Healthcare Ministry, in September 2009, what we've done is that we've, for the first time, introduced a definition of what a biosimilar product is in the Health Code of the Russian Federation, and that allowed us to continue to develop requirements towards these products and their registration. So here in the slide you see the definition that we have. This explains what a biological medical product is and what a biosimilar product is. Well, of course, this definition, if you look closely, is a bit of a gibberish, and we acknowledge that thing, but back in the day it was necessary, and this is what we've adopted. We've studied some of the best practices from the European Union. We looked at the WHO, and we decided to go with this definition. But if you look at the latest definition of what a biosimilar product is and read it very carefully, you see that it mostly relates to the so-called hybrid technology, genetic engineering, the mostly recombinant molecules, synthetically created molecules. Now, the requirements towards dossier. And of course, since we have a definition, we have to take a step further. Therefore, we have the requirements in terms of the originator's dossier. So here we don't differentiate between chemical or biological substances. We have one uniform body of standards in terms of dossier, be it chemical, be it biological. So, and this is a full and complete registration dossier. Now, the requirements towards the dossier for the registration of biosimilars. Let's talk about that one. And I'm giving you a very brief version. We have, like I said, the official law, 735, and the process starts from the very beginning and involves comparative studies for the product. The substance itself involves comparison products and comparison studies on a preclinical level and clinical level. And also, of course, as you understand, it involves the pharmacovigilance master file as well as risk management plan. Let me also mention that as of May 10th, 2003, we've registered more than 7,000 of these products, and that's excluding medical tools and equipment. So I would say that biological products would be about 300 of those. So we already set out the classes of biological products that we've registered. And here I'd like to share our experience with regards to expertise and of those biological products that were set out as biosimilars. Of course, we have very limited experience in that, as you probably understand. So, starting from the enactment of this law, like I said, we've started the process, and all these products have been denied registration for the following reasons. To begin with, if you look at the registration dossier of these products, there was no proof whatsoever about the similarity in terms of quality with the originator. There was also lacking clinical or preclinical data. They were insufficient or absent as such. Also, in some cases, the claimant would actually use various comparative assets during different stages. Sometimes the developer would use, for instance, the originator product, or for preclinical studies, the same producer would be guided by a totally different product, a product that was designed and developed by the same country but a different manufacturer. And that is absolutely unacceptable. So this is not what we can consider as a biosimilar product. So therefore, the product is void, and I would never register that, because there's no risk control plan whatsoever in terms of its harmful impact. So that's what we've received in 2013, that these products are currently going through the registration stage. We don't know, the decisions for them are pending. Now, defining the reference product. Of course, in terms of Kazakhstan, we believe that this product should be the originator. This is what we call it, and that's why we have the definition there. You see, this biosimilar, we have the reference, words like originator and all that. That means that the comparative product should be the originating product and nothing else. If the originating product is not registered yet in our country, so therefore, in that case, we have to look elsewhere. First of all, we reference to the European Union and try to find this registration, this product, somewhere in the European Union. Now, here I would like to give you the overall layout. This is something that you know. And since we have clear rules with regards to registration dossier, and these are quite general actually in terms of approach, and we're lacking specific documents that are describing in greater details the way the procedure for this registration should look like and the way you should assess this or that class of biological products, since we don't have that at this moment of time. Of course, we try to be guided by the WHO primarily and the European Union. We're currently working, of course, on national standards, and we're primarily guided by the above standards. And I believe there's something like in 2014, we should be having our own national legislation and national guidelines for these products. So my colleagues have mentioned some of the products that were registered either domestically or somewhere internationally before the official biosimilar product registration requirement took place. And we have such cases as well, such as the insulin-related product. Certain manufacturers of insulin products that were registered prior to this law, and we can't call these products biosimilars, but the thing is that the registration is now void, they've expired, and some of these are no longer available on the market. Yes, they're registered, but they're not circulating, they're not commercially viable, they're not popular due to the fact that, well, I would say it has to do with the procurement approach. I mean, the government procurement policy is such that it was based on the biosimilar requirements. So these groups of products weren't very popular in terms of the clinical applications and gradually were removed from the market. So based on this experience, there's only one case study that we're now trying to implement in legislation. And I'm talking about the product that is registered and is claimed as a biosimilar before the official requirement, and then you need to properly register this product. It's not a re-registration. It's a brand new registration where we look at this biosimilar product, if it's a such one, from the viewpoint of the new legislation. This is, by the way, a canyon, beautiful place. We welcome you to Almaty. Visit this place, it's amazing. Thank you. Good afternoon, ladies and gentlemen. Let me introduce myself. I'm Adrian Nadjaryan. I head the group of pharmacological experts. I'm a physician myself. So we analyze dossiers from the viewpoint of efficacy and safety. So I will be dealing with these items specifically because this is my regular activity. Let me look at the items that we look at, and let me tell you how we're doing. In the Republic of Belarus, you know that we have the official law on medications. We have the official definition of what a medication is. This definition, I'm not going to give you full definition, but I would say that it's quite special. It is actually a combination of existing substances, and any combination of existing substances is also a medication, so which calls for a lot of creativity. Arbidol with any other product can be a medication, so you can register dozens of new medications by simple combination. We also have the generic definition, and you know what generic is. I'm not going to give you the details of what it is, and I would say it's no different. It's a general description of generic. I would say nothing unusual, so nothing to mention here. We're currently considering a new law on medications, and it's actually going through two stages. It's been approved during the first stage. So, and like I said, with this new law, we're introducing a new knowledge, the new definition of what a biological product is, as well as the biosimilar product. And as you understand, we're introducing this definition because we already have amendments to these laws that deal with specifically these types of products. Let me also add the following here. The only proper law that we're guided by is Instruction 52 by the Ministry of Healthcare of Belarusian Republic that sets out the list of documents for submission and registration of a biosimilar product in our republic or any biotech product. So what it needs is a full preclinical study results, also clinical studies results that were done according to the best practices or the good medical practices as we call them. Of course, if the company can also support it with secondary tertiary data on the structure of the product, in that case that will be of course considered as an advantage. As a matter of fact, as a rule, we consider every biosimilar related product dossier in our own special way, and we set out specific requirements, predominantly guided by the European Union. Well, let me give you specific examples. I'm not going to give you generalistic statements, but let me rather take you to a specific step-by-step process. So, like I said, this is the main page, and you have that. This is the guidelines, mostly guided by the European Medical Products Agency. These guidelines were translated into the Russian language and they became part of our national legislation so that we can actually officially refer to them. They're part of our national regulation. So let me give you specific examples. This is my expert view, expert statement on a low molecular weight heparin product. And that was actually submitted for registration, and I'm not going to give you the beneficiaries and the brand names. It's not the setting to do that. Usually, our statements are quite fast. I would say that including the primary registration takes about 20 or 30 pages. If we have anything to add, then we write the full guideline, the full instruction, fully blown, and that's a lot of pages. Probably that's extra paperwork, but like I said, we have domestic manufacturers, and these companies actually force us to do all that. We're obligated to do this extra work. Okay, so this is the what you see on the slide is the beginning part. So what indirect properties actually allow you to get a clue about the quality of the product, such as this low molecular weight heparin. It's part of the GMP base, it's part of the FDA database, so that means that the product fully passed that and is fully certified. Well, these are indirect properties, but that actually is a clue to the quality. That means that you actually deal with the substance, that it has a prior track record, because in most cases you have nothing like that. You have empty space. Now, first of all, what did the company provide us with in terms of preclinical data? So they've given us some evidence of biosimilarity of both physical and chemical properties. They've also showed us similarity in terms of the raw materials, in terms of the molecular structure, and in terms of the biochemical and biological properties and studies. This is what they've demonstrated. So we counterattacked on the preclinical studies. We claimed that we're lacking comparative in vitro studies. There's no toxicity data and immunogenicity data in animals. This is what we've claimed, specifically for that process. This is for the preclinical trials. In terms of the clinical trials, also pharmacokinetic comparative studies, pharmacodynamic studies weren't done properly. The biosimilarity was not disclosed properly. I'm sorry, I'm completely mixed up here. This is what they've provided us with. This is what they've provided us with. So what they've done is they did pharmacokinetics, pharmacodynamics on healthy volunteers. And that's all we had, and nothing else. The thing about this is that the company actually did it according to the low molecular weight heparin product, according to the FDA approach and pathway. But the thing is that the FDA never considers such product as low molecular weight heparin as a biosimilar product. So FDA only requires a proof of structure similarity, but it doesn't require competitive, let's say, clinical and preclinical trials. So with us, the requirements take us further. And this is what we set out as a requirement for the company. So these were my claims on the clinical trials. I said that I'm sorry that you're actually not showing us therapeutical bioequivalence. You're not showing us any evidence of, the patients that have a higher risk of thrombosis. And what you've shown us are studies in a general framework, but you didn't give us details about any studies related to immunogenicity. Now, in terms of immunogenicity, of course, we're guided by the European requirements, and we use it. It's translated into Russian, and it's part of the national legislation. And we apply this concept to biosimilar products. Besides that, another requirement that we put forward is about this official medical instruction, that is the guidebook for the regional product. And we want this to be word by word. So we're not looking at the Russian documents, but we're looking at the official document which is approved by the originator country. So if the product originated elsewhere, we'll be guided by that local jurisdiction rather than the application country's code. Now, in terms of registering this low molecular weight heparin, because there was no comparison done, there's no comparative preclinical and clinical studies done, all you can say is that, well, you can only do it for hemodialysis for elderly patients above 70 years of age. This is the only area of applications that we allowed this product for. Other than that, this product was banned. The thing is that there is this specifics. Whenever you register a biosimilar product, or whatever you hold a clinical or preclinical trial, you have to follow a particular procedure. If this manufacturer would do the studies across the board, like a local study, and you don't need a lot of subjects, about 30 patients will be more than enough. 15 to 15. I mean, 2 groups. And this product would have been registered across the board, and just like the originator product. But unfortunately, they didn't do it. They didn't hold these trials. So that's why we only registered one side of the product. Now, the other thing I would like to mention here, and that is very important, if that was a domestic product being registered, of course, we would have been much more flexible, definitely. There is, of course, a protection exercise. We support local manufacturers. It's part of a national policy, but it was not a local product. Especially with the biosimilar products, we don't see anything like that. We don't see a lot of that being registered. Some attempts, some feeble attempts, but we don't see these products ending up on the markets and the shelves. So finally, of course, we're guided by this FDA pyramid. They have very nice biosimilar products. The pyramid, pretty much, I think, seen it many times. Now, what else? What else can I point out here? Another specialty that we characterize by is that quite recently there's been a number of attempts to register products that are classified as biosimilars from Cuba. The dossiers from Cuba, of course, were quite short. They had limited information. There was no comparative preclinical and clinical studies, and we didn't register these products. And the thing is that over the last year, what we've done is we had about 12 biosimilar products submitted for registration, and only 2 of them were partially registered. I mean, like I said, that low molecular weight heparin, and another one is from Korea. I think it was called Efeximab. That's the name of the product, and it received positive statement from us because the dossier was more or less, and like I'm quite a strong professional, and I've seen a lot of dossiers, and this Korean dossier was quite decent, quite strong, so we decided to register that product. But again, these are, I would say, mostly guided by FDA. Like I say, according to FDA, you need a full match. We do it in English, and then we do it in translation to the Russian language. So the structure of these registered biosimilar products, if you look at the way they were structured, there are quite a few, but these 2 are mostly interferon-related products. Right, and we're fighting this adverse interferon products. More or less, the market, I would say, is quite limited. We're not seeing a lot of biosimilar products on the market. So we know all the requirements, we know the American requirements and all the details, we know European requirements and all the details. But it's about choice. What are you ultimately guided by? The first or the second? Or take the European requirements, for instance. Should we be guided by these requirements in all of its scope? Should we indeed close our market to this low molecular weight heparin? Because this product is registered in South Americas, also Eastern Europe, Oriental markets, etc. Should we be that strict? Should we be guided specifically by the European requirements or not? I'm still struggling with that. I mean, are we being perhaps too strict with that? So I'm still undecided myself. So we're working, we're seeing as it goes, but perhaps, I don't know, probably the government will give us a clue, and some of these requirements could become more liberal in the future. Or probably we'll come up with our own products made domestically in Belarus. Well, we'll see how it goes in the future. That's all I wanted to say. Thank you. The title of my presentation is Regulatory Landscape for Biotech Products and Biosimilars in Ukraine, and the recent changes that happened to the requirements of the registration dossier, the evaluation and expertise. As you know, in Ukraine there is a state program underway to harmonize the regulatory basis of Ukraine to the official requirements of the European Community, the European institutions, and the European Union. At the moment, the use of medicines is regulated by the main law of Ukraine, which serves as the basic document, and that has already been harmonized and currently matches the requirements of the EU requirement and directives of 2001. But our task today is to bring to conformity, that is, to align this document with all other documents, such as the bylaws, the ordinances, the instructions of the cabinet, and the ministries of healthcare regulation, as well as align with all other regulations that are pertaining to this area. So the main laws, the main documents that regulate the market, the circulation, the sales and purchase of drugs in Ukraine, anything that has to do with medicals, is the official decree number 376, which states the official procedure of the state registration and re-registration, as well as law number 373, which ensures the official procedure and brings to conformity the requirements with regards to immunological products and harmonizes it with both national and international standards. And there's also the resolution of SMU of 2005 on the official state quality control procedure for medical products in Ukraine. Now, in order to practically implement these steps with regards to expertise, registration of medicines, and the like, the Ministry of Healthcare issued its all orders and guidebooks that give a detailed step-by-step procedure for these products. Up until now, there's been two orders in effect. Number 426, which is for medical materials registration, and another one, which is number 486, which stipulates that the official state control over the quality of immunological medicines, which is currently used in practice in Ukraine. Now, what are the key items here, the key requirements in terms of registering medicines today? In 2012, there's been a change, and I mean the amendments to the official ordinance of 717 of the government, which sets out the official regulation procedure for medicines as well as biological compounds alongside a unified procedure, because before that we had two procedures. We had medicines on the one hand, and we had biological products on the other hand, and they were registered differently. Now, starting from April 15th, we only have one procedure. And the effect of the state of regulation on immunological medicines is confirmed by a registration certificate and also a seal that sits on the medicine. I would like to also underline the fact that with this regulation, the Ministry of Healthcare now vested the right to actually terminate any circulation of any drugs in Ukraine that have not been imported to the country over the past 2 years, that this has not been in use. With that, we would like to exclude all sorts of situations where there is a manufacturer that would register the product for the sake of registration and for different aims, for political reasons, for competitive reasons, for whatever reasons, but don't actually import the medicine into the country. In January of this year, the Ministry of Healthcare issued a new order which introduced a dramatic new change to the main ordinance of the medicine registrations. And the changes were as follows. The definitions were given differently. Also, there was unification of procedures provided for pretty much all medicines, all medical products. The role of the Ministry of Healthcare was reinforced as the main regulatory body. And there was also a streamlining of the registration procedure in overall. Now, the following sections were also changed, such as sections regarding changes to the registration process. There was now a new changes to the classification. And another very important point was very interesting. It is now obligatory when you submit a document to provide also a copy of a document that confirms that you actually comply as a manufacturer to the existing requirements towards the GMP, Good Manufacturing Practices, and that has to do with both foreign and domestic producers and importers. Another thing that was reworked is Article 12 of the federal law dealing with labeling. There were other conditions that were also strengthened and became more stringent, such as the timeline of registration. Now, by the very same order, a more specific requirements were defined towards the common drugs. Were also definitions given to the criteria of conducting additional standards and research on medicines that deal with pharmaceutical development. Also, confirmation of quality of the medicine, studies of safety, efficacy, and the centers for equivalence and availability of these medicines. Now, the requirements towards the bioequivalence, and I'm not going to go into this, these are generics. I think that I'd rather touch upon the procedures that deal with the proper registration procedures for biosimilar products. The very same order provides specific procedures when you register original medicines that were on the official list of WHO and used for treatment of socially dangerous diseases such as the tuberculosis, HIV, hepatitis, all sorts of virus-related diseases, etc. And according to the order, there is Module 1 and Module 2, and you can submit that registration in any language, be it Ukrainian or Russian on a hard copy. Other modules 3, 4, and 5 can be actually submitted on any carrier. And for these medicines, there's also a fast-tracked expertise procedure and registration procedure, and there's a maximum term of 90 days. So that is the official time. So for the drug must be registered within that time. Well, I will skip some of these and let us better say something about the biosimilar products. Now, in Ukraine, biosimilar products include a whole range of products such as allergens, antigens, vaccines, cytogens, even modulators, medicines manufactured based on tissues, plasma, and blood of bodies and animals, interferons, and medicines that are used to treat specific immune-related diseases. Now, and these products can be obtained by stems of microorganisms, cell lines, eukaryotic lines, extractions from biological products, human plasma, blood, animal, plants, etc., etc. And the order sets forth very specifically what a biosimilar product actually is and what biosimilar medicines are. So this is a biological medicine. I'm trying to find here the definition. Give me a second. Yes, so this is a biological medicine that is similar in terms of efficacy, safety, and quality to a similar registered biological product, which patent has expired. And the similarity of the therapeutical efficacy should be confirmed with respective preclinical and clinical studies. That's what it says here. And there are specific requirements set out towards the dossiers of such biosimilars. And in the case of registration of such a biosimilar product, except for modules 1 and 2, additional data must also be provided to support the appropriate level of both safety and efficacy of such products. Now, the volume of additional trials is to be defined separately on a case-by-case basis. Now, there are also special requirements towards the registration of a dossier for biosimilars, such as for modules 4 and 5. You need to confirm the quality, the safety, and the efficacy for each of the indications. That is what's important, that you do it for each and every indication. Of course, if there's more than one indication, contains. So confirmation has to be done for each confirmation. Well, since I only had 10 minutes, I think that I will skip the government pharmacovigilance control. So, that's it. The only thing I would like to say that until recently, both the input control of all immunological products in the country was done by the government expert center who had a laboratory, and it was certified by the European Union. But according to the new innovation, as it usually happens in our country, the decision was made 2 months ago that this work will be done now by governmental medicinal service. That means that the control and the registration procedures will be separated. So they're divided now, and we're trying to combine it together. So all divisions and all departments are, of course, taking every step to complete the development of the official standard and procedures for each kind of examination. Now, this information is available on the website of the center. It is open, and we welcome you. Read and please introduce changes, participate in this process, be interactive. Thank you very much, and I'm open to questions. Thank you, Yuri. Ivan is really worried that we're not getting a full discussion here, because at 12 we're going to start a new session, and there will be some minor changes to the agenda announced. But we work today not according to European norms, but according to Russian norms, and that is from 8 a.m. to 8 p.m. So I think that the questions that Ivana has outlined for this discussion, these should be our homework for today. We'll think it over throughout the day, and you are free to ask these questions in any session. For instance, let's take a question that was addressed to me. And the question was, should we perhaps use the experience of the Republic of Kazakhstan and Belarus when we register and re-register biological products, including biosimilar products, pharmacovigilance, and dossier issues related? Well, the session that starts at 12 will answer these questions. And the answer is, in a nutshell, not only should, but will be. Because we exist in the same custom space, and any medical products and their circulation, any medical tools and biological products is an item that will be specifically approved in an agreement that should be signed, I think, end of May in Astana. So, and this approach assumes very common principles, unified approach to regulation of these processes. So the next session will discuss that, but I'd love to ask Ivana this question. Ivana was such a great moderator today, and I do know that she was very well familiar with the format of this discussion, and she was really willing to participate in this discussion. So Ivana, the question was as follows. In what WHO document can we find the maximum deviation between a biosimilar product and a reference product? Well, the original question says a reproduced drug, but I believe that it wasn't about a reproduced drug, because as Ivana said, that biosimilar product is never positioned as a reproducible drug. So it's not a generic. We're speaking about the difference between a biosimilar and the original product. So what would be the maximum deviation, or the margin as we call it? How far off should a biosimilar come to a original drug to be called biosimilar? So that will be the question to you. Good morning, dear colleagues, ladies and gentlemen. I'm your colleague, and I actually Russian, but I work in the United Kingdom for the state body MHRA. and I've been a medical assessor for biological products for the past 5 years. Also, I'm working with biosimilar products as well as other biologicals. So to continue my presentation, I will switch on to English now, because everything that I've done, all my work, all been tested over time in English.
This presentation is given in personal capacity, so it doesn't represent necessarily full views of FDA and European Medicines Agency, and also MHRA, but it has reference to appropriate guidelines and appropriate documents which we issued. There is no confidential data disclosed here, and all relevant references available from public domain. Now I'd like to begin with some facts, and you probably know that biologics market, and in this presentation I'll be focusing on monoclonal antibodies of originator level, so we're not going to be talking about biosimilars in this presentation, but I'll be covering that at the session this afternoon. The dynamics is extremely active, and we have a lot of products in phase 3, phase 2 studies, with almost 270 products worldwide in development. So this product's been tested through different scientific advice interactions with both FDA and European Medicines Agency, and mainly they're developed in autoimmunity, oncology indications, but increasingly we have some of these products developed for lifestyle diseases like bone disorders, for example, obesity, problems with cardiovascular conditions, and so on. So these monoclonal antibodies are coming in years for almost virtually all indications which affect human beings. The total production is also going up, and there is a trend that biologics will become commodity products in next 10-15 years. What means that with quality of production and improved cell culture facilities, we expect that by 2020 we will produce almost 80 tons of biological products, and about 10% of those will be biosimilars. From European assessment of biological products, first of all, I would like to say that there are products which have been licensed, approved in Europe and national countries, national competent authorities in times before European Medicines Agency was created through different routes like vaccines and, for example, blood products. But with creation of EMA, we have now mandatory route for centralized procedure utilized for recombinant biotechnology products, gene therapy, orphan products, generic versions of centralized approved biological products and chemicals, and also products like biosimilars and products important for European healthcare, such as infectious diseases like HIV or cancer. Just to explain very briefly how European procedure operates, we have CGMP, which is an organization within EMA, which is composed of experts from 27 countries of European members, and 2 rapporteurs assigned for centralized procedure, which work independently to come with a decision at day 80. And evaluation team then gathers through peer review process, which is third country, which then helps 2 countries to find consensus about various disagreements arising through the assessment. And then CGMP gathers at day 120 to sum up views, not just from these 2 countries, but also from all 27 members. And if necessary, throughout centralized procedure, we utilize expert advisory opinions from eminent experts in the field from different countries. EMA usually serves administrative function with helping interaction between the CGMP and company and requiring necessary documents and serving with appropriate functions. This is an outline of centralized procedure. So it's quite lengthy. It takes normally, as we always tell companies, around 14 to 16 months. And this dotted line in the middle represents the so-called clock stop period. So after assessment at day 120, company receives a long list of questions on quality, clinical and non-clinical issues, and company has time up to 6 months if necessary to address some of these issues and come back with responses. And then procedure restarts, and usually if there are issues which are not resolved, at the last phase of procedure we give chance to the company to come for oral explanation at CGMP, and then if product gets approved or withdrawn, we have public assessment report to ensure transparency around our assessment, and we have label issued throughout countries. As you know, with therapeutic antibodies, we have now lots of different products approved in last 15 years. But now the area is moving rapidly into so-called new classes of antibodies, which increasingly require lots of scientific expertise and understanding of how they function and what type of values they create to patients. So, apart from some of these typical monoclonal antibodies, as you know, created by FAB and FC fragments, we now have diabodies, for example, monovalent fragments of antibodies, and bispecific antibodies, which have ability to bind to two different antigens and have very powerful efficacy in oncology, for example. So in order to understand some of this dynamics on the market, we generally follow a principle that yes, there is a nomenclature of behind some of these monoclonal antibodies, and the principle in the past was to develop products which effectively fully human, so there are no sequences belong to mouse which originally been introduced or used because of hybridoma technology. And the principle behind to ensure that there is no immunogenicity. Therapeutic indications, as I mentioned, cover mainly autoimmune diseases like rheumatoid arthritis, for Humira, for infliximab, and Remicade, or Remicade and MabThera. Oncology, which is for Avastin, Erbitux, Herceptin, and recently approved product Perjeta and Adcetris. And also we have, to less extent, developed and licensed in Europe monoclonal antibodies for some of the infectious diseases like Synagis for respiratory syncytial virus in children. And last few years there was a product called Denosumab from Amgen, which was approved in two indications in two forms. One is for postmenopausal osteoporosis in women, called Prolia, and in different dose for different formulation. It's been also approved for skeletal related events in multiple malignancies affecting bones. Now also we've been involved in scientific advice for companies developing monoclonal antibodies for cardiovascular indications like canakinumab from Novartis, Alzheimer's disease from, for example, Roche and Genentech, neurology indications, muscle wasting, and problems of old age. So as I said, in years to come we expect some of these monoclonal antibodies to enter various types of healthcare. One of the examples which we've been dealing in last year for the biotherapeutics of new class include antibody-drug conjugates. Those actually developed on the basis of prior available antibodies which been linked with powerful toxins, allowing for some of these antibodies not just bind specifically to tumor cells, but also inject toxin directly into the tumor cells, enhancing efficacy many-fold times. And one of the examples is Adcetris, which is developed by Takeda Millennium and approved in Europe for the for indication of Hodgkin's lymphoma and also for the anaplastic cell lymphoma. And one of the products which currently under assessment and already approved by FDA is a product called trastuzumab DM1, which is a complex of trastuzumab with toxin allowing to enhance efficacy in breast cancer. So from quality perspective, and I would like to leave most of the expertise around quality to my colleagues here at the table, we use normally ICH principles, and there are a very good foundation laid into guidelines on development and production, characterization and specifications of monoclonal antibodies and related products issued in 2007. So that covers different aspects of quality. And characterization of all products when they come in for assessment is quite complex. So we require multiple methodologies used for companies to demonstrate they understand their product very well. So there are lots of different things which they can do, from proving to us that there is identity of the product based on physicochemical characteristics, sequencing, and using various other methodologies which are listed here. From non-clinical perspective, with biotherapeutics, we generally require some basic foundations to understand two issues. One is pharmacology of the product, and secondly, immunogenicity. And non-clinical studies can help to realize whether there is some phenomena behind exaggregated pharmacology, and also understanding of impurity profile. The typical requirements from Europe that we do not demand for monoclonal antibodies and therapeutics, for example, ECG or QT prolongation studies, and we require certain number of toxicology studies, but normally genotoxicity and carcinogenicity studies are not required. And reproductive toxicity data might be required on a case-by-case basis. For example, like with belimumab, which is a GSK product for SLE, since lupus is primarily disease in women of young reproductive age, this data would be useful and might be required. What is very important that, as you probably know, in 2007 we had 5 subjects in a famous TGN-1412 trial, which became victim of a very powerful monoclonal antibody which was tested in healthy volunteers. And from that, lessons been learned that non-clinical data with powerful monoclonal antibodies may not translate in optimal dose defined in humans unless there is a special criteria called minimally anticipated biological effect level, called MABEL, which is actually allows to demonstrate the projected occupancy of receptors with monoclonal antibodies. And now we recommend to use MABEL for some of these high-risk monoclonal antibody products. So MABEL is usually derived not using non-clinical animal data, but using human primary cell cultures or immunocells isolated in vitro, and by mixing antibodies at various dilutions with, for example, mononuclear cells derived from humans, you can see whether they release a lot of cytokines and various other factors. So then you can predict at what the optimal dilution you may start your dose in man. The clinical issues, first of all, there is no specific guideline which defines clinical requirements for originator monoclonal antibodies in Europe. So principle is laid in different guidelines which are defined for different therapeutic areas. And actually clinical development of these biologics is not different to small chemical drugs. So if, for example, somebody wants to develop monoclonal antibody for multiple sclerosis or for rheumatoid arthritis, we have guidelines for products for development in those areas. So they would need to address the principle of their phase 3 trial design using, for example, guideline for new products for rheumatoid arthritis or for multiple sclerosis and so on. We also encourage companies to come for earlier pre-phase 2, pre-phase 3 scientific advice at CGMP level, but companies also have option to come to national authorities like UK, Germany, and most important players within Europe to actually address issues they may want to discuss face-to-face. And general principles that there is a case-by-case basis based on therapeutic indication, patient population, route of administration for this product, usually parenteral. And we now encourage companies to think about biomarkers and biomarker stratified defined indications, which actually will ease to make a judgment about benefit risk for these products. And companion diagnostic tests should be developed and validated, ideally through phase 3. From at the point of assessment of the efficacy data with these products, we usually look at the magnitude of the clinical effect and how meaningful this effect to patients and prescribers. So for example, if we look at the overall survival, we usually make a judgment how many months in addition of median survival it gives, and whether this survival increase was actually accompanied with good quality of life in patients. Any adverse events which occur during that time which might be detrimental to patients. The type of clinical benefits also very important. If there is a new product for rheumatoid arthritis, we'll look at also for new products with radiographic evidence for support of structural effect. Whilst with biosimilars, this data get extrapolated from reference products. So there is a major difference here. Effect on quality of life and various outcomes like survival is important, and also, as I mentioned, about role of biomarkers. Immunogenicity is a critical issue now, and for new products, the risk is always high. So we expect the assay by the company to be developed from phase 1 onwards. There should be risk-driven approach with biologics, and generally assay should be validated in terms of sensitivity and specificity. There should be a justification how periodically a specimen's been collected in a trial, and also whether the immunogenicity actually accompanied with some adverse events resulted from this problem. And monitoring ideally should be done up to 12 months. But we do not require, compared to FDA, statistical hypothesis behind immunogenicity. So descriptive statistics is usually sufficient. As you know, there are examples of how monoclonal antibodies can generate quite serious problems with immunogenicity. And with products like adalimumab or infliximab, immunogenicity can reach quite high level in patients. So those patients may either lose response to the product or have some various adverse events. And generally, deficiencies we find in dossiers when we assess them is usually either assays are not sensitive enough, so they show zero immunogenicity, fantastic, but that doesn't mean that there is no immunogenicity. So we ask to develop another assay or re-assay all specimens based more sensitive assay, which will show whether there is actually some immunogenicity underlying it or not. The lack of interpretation of data, so if there is immunogenicity, what does it mean? So we would like to see efficacy in those patients on individual level, look at how this immunogenicity translates into safety. And some companies have been able and been smart to actually use immunogenicity in post-marketing period, like with Remicade, to tailor management of patients throughout their treatment. From safety point of view, with all biologics, we expect risk management plan. It's mandatory in Europe, and with obviously this pharmacovigilance area is particular concern for monoclonal antibodies. There are some serious and poorly understood problems like infusion reactions, opportunistic infections, and progressive multiple leukoencephalopathy, which is actually very serious and potentially fatal serious adverse event, is reported with products like rituximab, natalizumab, and efalizumab. Also, cancer has been reported with lymphoma, with adalimumab and infliximab. And recently approved denosumab actually has quite important and very debilitating problem for patients called osteonecrosis of the jaw, which basically results in collapse of the jaw. And patients with cancer who treated with this product may have not been able to actually live longer because of the fundamental issues arising from this phenomenon. So safety of biologics is actually a very complex issue, but in terms of making a judgment about risk-benefit balance, what we would like to see is this benefit net between number needed to treat, which means how many patients you need to treat to actually treat or cure one patient. And with infliximab, for example, is 1.5 patients, which means you need to treat 1.5 patients to actually achieve effect in one, which is very impressive. And on another hand, in order to get one case of tuberculosis with infliximab, according to statistics, you need to have 681 patients, which is called number needed to harm. So obviously that gives you a flavor how benefit-risk judgment is made. So because of the rarity of the serious adverse events and way how they can be controlled and very high level of benefit, we can make a positive recommendation for such product to go on the market. And risk management plan for the biologics is usually include routine passive surveillance, which might be sufficient for most of cases. So looking basically what signals emergence throughout the post-marketing life of the product. We also expect that it will be education of prescribers and patients. So patients might need to have alert card, which will be available if something will happen in emergency. So prescriber will know or physician will know that they taken rituximab or adalimumab. And that's important. And finally, in some cases, proactive surveillance is necessary in form of registries or post-marketing safety studies, as has been done already with Remicade, Humira, and Cimzia. Pregnancy registries are also very helpful because they can help to shape label in post-marketing phase of the product. One of the products which is actually shows example of how serious risk has been dealt with and very well managed is natalizumab or Tysabri, which has been approved in Europe for relapsing forms of multiple sclerosis, and FDA approved it for both MS and also Crohn's disease. This product induces a very serious fatal problem, which basically results in dissolution of the brain, and people develop manifestations which are very similar to stroke. There are currently about 150 cases of PML described worldwide, and PML can be fatal in almost 20%. The current risk management plan for natalizumab allowed to control and reduce risk of PML using, first of all, very restricted prescribing of the product amongst specialists in multiple sclerosis who understand PML and who understand how to use natalizumab, but also a company developed specific serology assay which has been introduced now into European and US label, and that allows to stratify risk and identify patients who are running at high risk of developing PML and remove those from initiation of therapy with Tysabri. Now, we also have this type of product dispensed through specific registry, and a company actually working with other companies in developing safety framework around PML, because effort of one company is often not sufficient to understand some of these very rare but very potentially fatal serious adverse events. So new European pharmacovigilance legislation was introduced from 2012, and there is a good emphasis there on biologics. So first of all, biologics and biosimilar will be scrutinized, and there will be definitely a requirement for intensive pharmacovigilance in first years of introducing those products on the market. There will also, in relation to interchangeability and switching issues, but also potential post-marketing problems, all members of states at national level recommended that all serious adverse events should be reported using the name of medicinal product and also batch number. So typical clinical issues arising during the during evaluation of dossiers with monoclonal antibodies is usually not sufficient demonstration of efficacy. So for example, very little increase in overall survival. We usually take this arbitrary approach, less than 2 months is not sufficient. So we're looking at more than 2 months of survival increase. We also look at the different type of risks which might be very difficult to control or predict. So for example, with products like Breakinumab from Abbott or anti-IL-12, there were issues with cardiovascular risks and also serious infections and malignancies which might not be compatible with psoriasis indication which this product was developed for. And certainly with Raptiva, which was ifilizumab, which had been withdrawn from the market in 2009, the issue was 4 cases of PML, which basically made an incidence of PML as 1 in 500, which was actually ahead of natalizumab. And this was indication in psoriasis again, which is not compatible with seriousness of such serious adverse events. So in conclusion, European regulations on novel monoclonal antibodies are mainly focused around quality issues. Non-clinical guidance is mainly drawn from ICH principles. Clinical guidances are products and group specific and shared with new chemical entities. Safety requirements are usually extensive, and safety databases are usually larger than with small molecule drugs. And risk management plans are mandatory, and enhanced post-marketing surveillance is something which we expect normally with this type of products. Thank you very much.
So, good afternoon. Looking here around, I think this room needs a bit more energy. You had some coffee before, now I'm standing between you and lunch, but there was some nice food outside there, and so I think you can stand it for another half an hour, at least I hope so. So actually, first of all, I would like to thank the organizers for inviting me to this, what I really think, very important conference here in Russia. It's really a pleasure for me being here. And actually, when being asked about what I may talk, I thought, you know, sometimes it's not so bad to make a step back and actually look where we came from. Where we came from and where do we stand now, and what eventually is necessary in order to maintain a certain level. And this is actually the presentation I have here. Here you see the outline. I will talk a bit about disease challenges, strategies and development around there. Then really looking back where we came from biotech and where we are now, have a very close look at regulations, and last but not least a couple of words also on biosimilars. Understanding the diseases. I think if you really look back, we during the last 100 years, we really made a great progress here. coming from an observational state, really having the patient in front of us, trying the one or the other media in order to provide cure. If this was not working, eventually we were trying another thing. We then learned what actually are the molecular basis of many diseases. When the DNA was discovered, when proteins were discovered, when we learned that for some proteins, when they are changing, we may have an issue. When they are overexpressed, we may have an issue. When they are underexpressed, we may have an issue, and so on. And today, actually, we understand even the more about the mechanism of these diseases. So we discovered the pathways for certain diseases. But the consequence of all of this is that more and more targets, in fact, we can have that are associated with a certain disease. And so the question really is, how can we find out the best target? What to tackle in order to provide cure? And in fact, this is getting more and more challenging. So in addition to that, we also learned that if a drug works in one patient, in one single patient, it does not necessarily mean it works in the other. So the challenge is to really identify the patient, the group of patients, the population in which a certain drug is going to work. Because otherwise, applying the wrong drug to a certain patient may even be quite harmful or in some cases fatal. We have made a lot of progress in this area. We can identify for certain therapies these days patient population and make these therapies extremely effective in this population. Sometimes we are even able to change the natural progression of a disease. Here you see really a nice example. A couple of years ago, when women were suffering from breast cancer and they had overexpression of HER2, this was a very poor diagnosis. Today, identifying this population, we really changed. Because today, if a woman has breast cancer and has overexpression HER2, this might be even a positive diagnosis given the highly effective treatment we can provide for that population. And this is exactly what we have to work on. This is what I truly call innovation. Now looking at the manufacturing process, looking at these products, where did we come from? Here you see quite a nice picture representing Emil von Behring when he is immunizing a horse. You know, biotech, pharmaceutical biotech started there, started in that area. But the actual, how can I say, revolution eventually started with these two guys when business met science and they formed one of the first biotech companies and really drove the manufacturing of these products. They really made it happen that we were able to modify animal, bacterial cells, that these cells in fact are making a human protein. But a protein? Not really. If we look in detail, and today we have more and more sophisticated analytical method, we really find out that we are not talking about a protein, a product. It's a highly complex mixture we are talking about. Given all the post-translational modification, given all the different degradation pathways these proteins may follow. We're talking about mixtures containing 100 millions of different molecular entities. And this is the true difference, also comparing small molecules with biologics. While a small molecule we can synthesize to a purity of 99.999%, with biologics we are always dealing with these complex mixtures. And we can characterize these mixtures to a certain extent and learn more about certain properties of certain species. This is how we were discovering the importance of glycosylation. Talking about a monoclonal antibody, minor changes in this glycosylation can have a tremendous impact on the profile of these, on the clinical profile of these antibodies. But we learned even more. We can shift a certain profile in this complex mixture. We can enrich the mixture with a certain profile, and this is going to allow us to engineer antibodies, for example, with certain molecular or certain mechanistical properties. We also can modify these antibodies. As we have heard before, we can couple toxins to them. We can shift the glycosylation pattern in one direction. We even can have monoclonal antibodies recognizing two different targets. This is what we call now bispecific antibodies, allowing completely new ways of treatment. One example we heard before is the coupling with a toxin, and here you see where you really can combine chemotherapy and immunotherapy with one compound actually, where the antibody is bringing the toxin to the cell, where the toxin is released in the lysosomal environment in the cell, but not outside, really pushing down all the side effects, all the adverse events you normally would observe when you apply chemotherapy only. What I also wanted to draw your attention, developing this program, currently and in the past, 14 clinical studies were ongoing, and this is just the start. Driving glycosylation in a certain direction, stimulating our own immune system, that this immune system is fighting cancer. This is what we are looking for when engineering the glycosylation of monoclonal antibodies and driving ADCC to a potency we never thought about before. Here another example for a monoclonal antibody that will be used in cancer indications. Also here, 10 clinical trials are ongoing, and still the product is not yet approved. I was mentioning clinical trials now couple of times, and there is a reason behind, because still many people think, okay, if you develop a new product, you make a phase 1 trial, you make a phase 2 trial, and you make a phase 3 trial, and then you are done. But this is no longer the case. More and more clinical trials have to be done in order to get these products approved, especially looking for different indication. Here, for example, for bevacizumab, only to give you an idea, 500 clinical trials have been performed with this compound. So really, this is a huge effort driving innovation here. And this is why, very clear, also innovation has a certain price. You have to invest more and more all the time. These drugs are not cheap. And this, of course, we need, therefore, also biosimilars, very clear. But safe and efficacious products. That brings me a bit to the regulatory part here. And I do not have a lot to say, because many of the things have just been said before. We were talking about ICH. This morning we were talking about WHO guidelines and so on. Well, the regulations actually are part of a whole environment here. These environments not only involve the regulators, it involves payers, but also investors. Do you want to invest your money in a company, or do you want to leave it in a bank? You know? I think it's very clear pharmaceutical industry has to make some money in order to drive this innovation you have seen before. But we also have to understand the concerns of the payers. There are cost pressures all over the place, and in between the regulators are sitting. Sometimes what we see, and I really have to say I do not like this a lot. Sometimes what I see when talking at conferences, you always see that there will be a mix-up between price and regulation. And many people think that by lowering the standard of regulation, you may drive access. But I think these are really completely two different issues. And one never should misuse a regulation to drive access. From my perspective, regulation, the task of the regulation is to ensure patient safety. Nothing more, but also nothing less. Now with regulation, I only have one slide. And actually, this I want to specifically tell to the regulators. What we really need is convergence. It doesn't make sense that each and every country is coming up with their own regulatory systems. This will drive industry nuts. It will actually block innovation at the end. What we really need is convergence. You have to come up with one nice and clean development program. You follow this, and then you really can get registration in many different countries. So really from an industry side, our wish very clear is remove, duplicate and different requirements which actually hinder this kind of innovative approaches. What we really need are new ways also how to regulate innovation. Let's talk about ways how to bring forward adaptive licensing approaches, how to move along breakthrough therapies, how can we provide access to new therapies more quickly to patients. This is actually what we need. Also, interagency reviews, when talking about the resource constraints we see in many agencies, these are things that should be discussed. If you think about many countries that have very small agencies, why can they not get together and review together a dossier, make a common decision on those? This really would help a lot and would really drive also the access. Also mutually recognize GMP inspection, an evergreening thing I'm mentioning here, but nevertheless I will mentioning it here. Does it really make sense to have one facility inspected by 20 different regulatory agencies? Does this add value? These are important things we really should discuss and not invest our resources in reinventing and reinventing and reinventing the wheel. Okay, now I stop. But no, I'm not yet finished with the topic. Because actually I think, now this brings me to the last part concerning biosimilars, because I think really WHO actually did a great job in bringing up these guideline on similar biotherapeutic products and are really taking a very important and I think also quite effective role in driving convergence with respect to biosimilar regulations. But I think we need much more of these kind of efforts. And also the EMA is really doing a great job. Today, before, we heard some thoughts around EMA regulation. But to me, one of the most important documents from EMA, and I strongly would recommend to everybody who is interested in biosimilar regulation to read this document, really is the guideline on biosimilar monoclonal antibody. I think EMA really was driving here the concept of similarity to the next level. Because here you see on this slide the main principles how to define similarity, which are nicely outlined in these guidelines. And for me, these are three main principles. First of all, we have to ensure that the previously proven safety and efficacy of the drug is conserved. This is what you want. If you remember one of the first slides on breast cancer, where I was saying we have changed the natural progression of the disease. We want that to be continued. We don't want to see a change in the slope of the curve here. This is why this principle is so important. The other principle, as well important, it is not necessary to prove again patient benefit. And this is new. This shows us really this is a completely new concept we have not seen before. And last but not least, all studies have to be done with the goal to detect potential differences between the reference product and the biosimilar candidate. So what I hear very often, we just do a clinical study. This is not the case. It has to be the right study. We are talking about targeted development. And what is the population we need in order to detect any differences? It has to be the most sensitive population. And this actually is what EMA is talking about in that guideline. And here on that slide, you actually have a comparison. What is the different approach, from the clinical side, comparing a biosimilar development, a true biosimilar development, and the development of a new biological entity. So you have to look for a highly sensitive patient population. You have to do comparative trials with the goal to show equivalence, not to look for superiority, what we are doing when driving innovation. The study endpoints have to be sensitive, and specifically when looking at oncology, these are not the classical endpoints. When you do an oncology trial, normally you look for overall survival, or at least for progression-free survival. These are not necessarily the endpoints that are used during a biosimilar development. The safety has to be comparative, and of course immunogenicity. And on this slide, for me, this is a very important thing, and you really should have in mind when discussing new regulations on biosimilar. The sensitive patient population for the required similarity assessment with respect to PK, PD, efficacy, safety, or immunogenicity might not be the same. So in other words, you may have to use a different population for PK, because what you want to find out in the study is, are there any differences between the biosimilar and the reference product with respect to PK? So what is the most sensitive population for that product that could detect differences in PK? In immunogenicity, you ask the question again, what is the most sensitive population to detect any potential differences with respect to immunogenicity? For sure, it will not be an immune-compromised patient population. You see, this is the principle of similarity. Asking again, what is the most sensitive population to get the answer to a certain clinical question and then go forward. Interestingly, in a recent paper, I only can recommend this paper to you, written by top EMA regulators. They are really making the point again. We are talking about highly complex mixtures. And not a single assessment, be it quality, be it safe, be it preclinical or clinical, is sufficient to show us similarity. They all have their gaps. And this is why we need all the three assessments in order to decide truly on similarity. And they clearly write a relaxation of these requirements is not justified. And therefore, to me, biosimilar, you can think about it like a house here. Unfortunately, we have an issue with the formatting here, but this is built on 5 pillars actually. We base similarity on the analytical comparability, on preclinical similarity, clinical similarity. A very important pillar is also the quality system of a company, because similarity shouldn't be a one-time event. You have to be sure that the company is capable on the long run to manufacture a safe and efficacious product, and last but not least, the pharmacovigilance. And with this, I like to thank you very much for your attention.
Okay, thank you very much.
So, my name is Alexander Dön from Biointegrated Company. My question is that in the magazine, the journal, Nature Biotechnology, a very authoritative journal, 2 years ago there was an article that the idea of this article was to analyze 3 biotech products on the market. One of them was by Roche, and that was Redoxon Maptera. And during this analysis, they've identified a dramatic change between batches in quality. And the chromatographic analysis, the biological activity, the potency, everything was different. Roche as a company didn't come back with any feedback, didn't make any statements on the market, didn't do any further research. And what's more, there was no messages from the market that these changes somehow had an impact on the treatment of patients. So my question to you would be, how would you comment on that? It turns out that it was insignificant, despite the fact that what underlined that the quality, the structure of the product, its potency, its efficacy has changed dramatically from batches to batches.
Your question, and
I think that was for me. No, first of all, thank you very much for the question, because this is something that is coming up over and over again. And let me explain 2 different things here. First of all, none of the variations that have been published there were going beyond our clinical experience with the product. Because normally what you have when you develop your product, you bring into the clinical trials quite a lot of different batches. So here you introduce a certain variability into the clinical study. And when you come then to the market, your manufacturing process is working in much smaller limits, okay? Because it's a large scale, highly controlled process. Now, if you make a change to this process, immediately this will be reflected in the product, and you will see some changes here. But then if you go back to your clinical experience, you realize, well, this change was covered with the clinical experience in the past. And this is exactly how the evaluation process of comparability works with the agency. So what you do here, you look at the differences you observe, you make a risk assessment, and this risk assessment will include your clinical experience, and then the agency comes to the conclusion, okay, we can justify these differences. So in reality, for regulators and for industry, this is nothing new, that you observe these kind of differences.
Thank you very much. My name is Anka Karatkova, Public Health Institute from the Ministry of Healthcare, Russian Federation. First of all, I'd like to thank you for two brilliant presentations. On the last slide of that presentation, you gave us some basics for the assessment of biosimilar products. Maybe I'm from a little bit of a different audience, because here in the room you mostly have pharma specialists. But still, it is quite surprising, and the fact itself that when we differentiate between a generic drug and the original drug that it was copied from, we always mention this issue of intellectual property, the patent protection. So there's a patent for the original drug, and then it expires, and then you can launch a generic drug on the market. But when we speak about biosimilar products, somehow we never mention IP. We speak about reproducibility, we speak about protection and safety. We always focus on similarity. But why nobody speaks about the intellectual property as a component of this formula? Why is it not part of the scope of discussion? Why is it that the safety becomes primary and the quality and reproducibility, but not the intellectual property?
There is absolutely, definitely emphasis on intellectual property as well. So I think there is an element of confusion here. So none of biosimilar products can enter on market in European Union or United States unless the patent for originator product will expire. So for example, if there is a biosimilar infliximab and patent will expire this year, the biosimilar infliximab may enter market only upon expiry of such patent. So of course there is also patent principles, but we are not looking into this when we do an assessment of efficacy and safety. So we're looking at the clinical implications for patients, prescribers, and for the product.
Actually, I received a question from Daria, and the question that Daria asks is about the monoclonal antibody production. Can you use the same cell line that has already been used to produce erythropoietin? So that means that you're basically using the same cell line, then validating and then purifying it. I'm not really a specialist in biological products manufacturing, but I think it's an extraordinary situation. And I think that, well, I personally, as a regulator, wouldn't let this pass. At least for the fact that I believe that there could be some added impurities in the mixture. Probably Carl knows better about it. Would you please comment on the situation?
Is it possible to use the same production cell line to produce two products, erythropoietin and monoclonal antibody?
From the technical point of view, I guess it could be possible. Why not? I mean, if you in the same cell line, you insert two different situations, the two different genes. Technically speaking, it's feasible, but I don't see exactly. I wasn't able to catch the real point. Maybe if you want.
Maybe I can add to this as well. Perhaps there is some confusion around. For both epoetin as well as a monoclonal antibody, you may use a CHO host cell, okay, as a starting material if you wish. And then you can transform the CHO host to either produce epoetin, or it also may produce a monoclonal antibody dependent on the gene you insert. The use of CHO is quite recommended for these processes because the glycosylation that is put onto the glycoprotein is quite similar to the one we find in humans. This is why people like to use CHO cells for these kind of processes.
Okay, I guess just one comment I would like to make concerning the last presentation. We had several times the request to get harmonized throughout Europe, throughout the world, and we are also trying to get to reach such an harmonization. But first, when we think about Europe, well, we understand that we also have to reach harmonization within Europe, which is not so easy. And my last point is that we also have some initiatives to get harmonized, like International Conference on Harmonization with Europe, United States, Japan. And we also had several other initiatives, but for some reasons they are not always reaching a good end, like the ICH for GMP inspection. I mean, the mutual recognition, which is not yet in place. So I will give the floor to you for the conclusion.